Welcome everybody. I'm super excited to welcome you all to the Yaya Foundation's 4-H research session at ULF. My name is Ron Garber. I'm the co-founder and board president of the Yaya Foundation, and it's super meaningful and exciting to be here with old friends and new. I'm going to start by repeating myself for those of you who were at the virtual care day that we did a couple of months ago, and share a story, because especially being here in person and virtually, this presents a tremendous opportunity for our disease community, and I want us to starting at this moment and for the next 4.5 hours, really try to make the most of it and seize every moment presented by this opportunity. And to sort of illustrate the opportunity, I'm going to go back to a story about 5 years ago. And again, for those of you at Virtual Care Day, you've heard this story, but we were in a room, the 4-H breakout room at ULF. It was a lot smaller room than this one, and there were a lot fewer people there. Which standing here now, I get the chills looking at how far we've come since then. But my daughter Yaya had passed away recently, and there were maybe 6 or 7 of us in that room, all tired, frustrated, sad at the lack of urgency and the lack of progress being made in our disease community. And we were frustrated. And Dr. Bernard was there with us. It's a great example of partnership between researchers and clinicians and patients. And she was frustrated. And we decided that we were going to do something about it. And we founded the Yaya Foundation coming out of that meeting. Today, the Yaya Foundation is the only organization in the world that's dedicated to fighting for people affected by 4-H leukodystrophy. Okay. And our mission is to provide educational and emotional support to people and families affected by 4-H, and to accelerate the discovery of therapies that will help improve the lives of people affected by 4-H. We've accomplished a lot since that meeting. We've launched a data collection program, which you're going to hear more about today. We've partnered with Dr. Bernard, Dr. Wolf, other folks here today to advance research and to advance, in particular, a transformational gene therapy project that we hope can be a building block. No, hey, Claudette. Hey, Claudette, it's great to see you. So we've launched our gene therapy project that we hope can be the building block for gene therapy for our disease community. And we've been recognized as a best-in-class rare disease patient organization by the Chan Zuckerberg Initiative, who gave us a prestigious Rare as One grant. So we've accomplished a lot since that day 5 years ago, and now we're here today. And I want to point out really that none of what we've accomplished in the last 5 years could have been done without partnership between researchers, clinicians, patients. And so it's meaningful that we're all here together in the same room. It's really meaningful that we have awesome researchers here today who have spent several years working on our disease and who are meeting our patient community for the first time, and who I hope will be really moved by these meetings and will go back and continue your work and work together with us and partner. I'm really moved by our team. And in fact, so I want to introduce our team quickly and then share for a minute about one of the things that I'm most proud of. But Christina Butterworth, our executive director, Valerie Greger, our research director, Shannon Healy, our advancement director, and I want to also introduce Lindsay, who's just joined us as a communications intern, who's going to help us share our story. This team is amazing, and I take really just great pride. One of the things I'm most proud of today is that each of them will be presenting on our data collection project, on our research strategy, on a needs assessment that is sourcing what is important for our families in terms of research. And, just 7 years ago, I had never heard of 4-H leukodystrophy. 3 years ago, none of the others had heard of 4-H leukodystrophy. And now these great people are really devoting their careers and their lives to helping our patient community. And so I'll conclude with a point that research can't be done without us. The reason I'm proud that our team is involved in research is presenting today is that research can't advance without patients and families. Our data is critical. Our stories are critical. We can bring awareness and funding to our disease. And so I'll conclude with that call to action that let's make the most of this day together, because research can't move forward without us. And with that, I'll welcome, I think, Christina to come up and take over. Thank you, everyone, and have a great, great day.
Everybody in, and it's such a great story. Thank you for that. So I won't belabor the agenda here. Oh, you need to be able to hear me, and I am a quiet talker, so I'll wear this. I won't belabor the agenda here, except to kind of orient everyone that we will spend the first majority of the time talking about research that is underway and any relevant, updates and things that we're really excited to share with you. And we're super excited about our guests who are both here in the room with us and joining us virtually. And then after a break, Valerie will really talk about, we've already shared a roadmap with you, but we are continuously learning. And so what have we learned since we last talked about the research roadmap with you? And really, really hope that there's some great discussion throughout, by the way. And then Shannon and I will talk through just all the non-research things. Like, we just really want to understand, where are you all in your journey? What is important to you? What are the gaps that we can help to support you? And there's so many people in the room that can, really enrich that conversation and help us think through what that looks like. So we're going to do our best to stay on time. And my team knows that I'm going to try really hard to do that, but here we are just a few minutes behind. So I'm going to dig in if that's okay with everyone. All right. So the first thing I wanted to just touch on, some of you are very familiar with, some of you have already participated in and engaged in, some of you are champions of, and others of you are new to it or just aren't sure. And so I wanted to just spend a little bit of time talking about what data collection is and how far we've come in the year since about a year since we launched, and then answer any questions. And the next thing on the agenda is natural history. And so those two things work really well together. So Dr. Nicole Wolf and Dr. Genevieve Bernard are here to sort of help continue that conversation as well. So as far as what data collection is, really at its core, it's really an attempt to capture the journeys that each individual is on. Because as we know, 4-H leukodystrophy does not look the same for anyone in this room, anyone on Zoom, or any family we have yet to meet. And so by bringing together our community and understanding I always call it like who's in the tent, right? Like who is impacted by 4-H leukodystrophy and how can we capture what that looks like is critically important to understanding the disease, to engaging with industry. We had a really great conversation last night about, there are plenty of organizations out there who could potentially help us, but who don't know anything about what 4-H leukodystrophy is. And so data collection is one of the means that we get to use to say, this is who we are. This is what this disease, starts to look like. And natural history is a very in-depth extension and continuation of that as well. So our goal at the Yaya Foundation, as you know, we're the only organization in the world focused solely on 4-H. Our goal is to build the most robust data collection. Our goal is to be able to go to industry partners and to be able to go to clinicians and other researchers and say, we are in touch with 100, 200, 300, whatever the number is, we are in touch with this many families. This many families are engaged in our community and want to tell their story and be a part of the solution, a part of finding the solution. And so we get really excited. I mean, I guess April, at our virtual care and disease management day, I think I shared this story with some of you when we were doing sort of like the warm-ups and the prep with some of the speakers, and we told people, you know, who was in our data collection, not who, it's de-identified data, but the numbers. I mean, their eyes got real big, you know. We've said a couple of times here, we're not messing around, you know, we're here to really make the big progress, start these conversations, and really start to continue to build on the momentum that many of you actually were the founders of. So, you know, I have a series of bullets here that you'll kind of, your eyes will gloss over, but really this is an opportunity for the patient voice to be heard. That's kind of the nutshell. And, you know, here's what we can talk about. The great thing about data collection is you get to go in, and when symptoms have changed, when you see something new, et cetera, this is sort of like at your discretion and at your experience that you get to contribute to the story. Dr. Nicole Wolf actually joined us on a family call last year, and so some of these, Nicole, will look familiar, but I love the AIDS clinicians and counseling individuals and families what to expect, right? So the more that we can share out with clinicians, particularly those who are seeing new patients or don't see many patients, the more we can sort of share what our community looks like and what their experiences are. And so you'll see that it's everything from, in this list, everything from, you know, clinical to partnership to research, to the time that we start to get really excited about moving closer and closer to clinical trials, like reducing some of the time or removing a barrier that, you know, we have a control group that are patients, because we can use data as a control group. So, and we've just identified in our own conversations, but also in further conversations, even last night, that really, you know, when we engage with potential industry partners, one of their very first questions is, do you have a registry? Do you have a data collection, otherwise known as a data collection? Do you have a natural history study? Do you know where your patients are? Do you have a global presence, or are you just in the United States? What are the age, you know, what do the age ranges look like? And all of those, some of, you know, they're trying to size up what the community looks like. And a data collection is a means to do that. So a little bit about how the Yaya Foundation went about this is we actually partnered with a nonprofit organization called RareX that was sort of started out of Empowered on the platform of the Broad Institute, which is a collaboration between Harvard and MIT. We were really excited at the idea that we were not going to set out to build our own data collection. We had heard from many patient organizations who had spent a lot of money and a lot of time, and maybe a decade later deciding that they had sort of done it wrong or had invested maybe in the wrong way. So the idea of partnering with an organization who sort of knew what they were doing and were thinking sort of pan disease was incredibly attractive to us. So we launched last summer, as I mentioned, and then additional surveys and functionality was rolled out at the very end of January. And we'll talk a little bit about what that latest release might mean in terms of action for those of you who've engaged. But the goal is really to, the other thing that was attractive about RareX was the sort of user interface. We didn't want to go with something that was going to be hours and hours of, you know, sitting down and uploading a bunch of files and things like that. So for those of you who've experienced it, it's sort of bite-sized in that you can start with sort of your general medical information, and based on how you answer those first surveys, you might be served up then like a deeper dive into a specific area. And so it's sort of like it's a very personalized journey. You're not asked to fill out, you know, seizure information if seizures are not a part of you or your child's journey. There will come a time when you can choose to upload your medical information if that is something that you are interested in sharing, though that is not a requirement right out of the gate. So it's not to say that that again, you need to have all the things, you need to have all the papers, and you need to commit to multiple hours. This is very like kind of at your pace and at what you are comfortable and willing to share. And for those of you, again, who've experienced it, there are multiple levels of what you want to share with whom. So you may choose to share your contact information with the Yaya Foundation, or you may not. You may choose to share it with pharma, or you may not. You may, so there's very distinct levels of data sharing, because we wanted that to be in the, we wanted you all to own your own data. But that last bullet there is really kind of the meaty one, is, we really wanted a secure environment. We wanted standardized data, again, not data that we just created that only we knew how to sort of, optimize and leverage. Robust in that, as we'll talk about in a minute, this is sitting on a platform that other diseases sit on as well. So this is not just 4-H leukodystrophy. There is another leukodystrophy community leveraging the same platform, and many other neurotype disease communities as well. So you can think about, we know that our community is rare and ultra rare, but when you think about the power of, our patient data, sort of, I like to think of it as like swimming in a much larger data set. And when you think about a broader set of researchers being able to see some of that de-identified data, our data could come up in a way that is, presented to a researcher who knows nothing about 4-H, but could lead to some really interesting collaboration and learnings and things like that. And so I'll just a really quick comment, and this, maybe I should have said, this is just a quick snapshot of what it looks like when you land at our homepage for the data collection. Lindsay, I think Shannon was gonna drop the URL in the chat for those of you online. And then this was just a quick snap to show you the number of new surveys that were released late January. But again, if you aren't impacted by, oral health, maybe not the best example, but, you wouldn't see those surveys. So I just wanted to sort of demonstrate that, their pipeline of what is continuing to be rolled out is quite significant, but again, personalized to your experience. So just a real quick snapshot of where we are today. In terms of the most recent data, in about 11 months, we have 65 individuals who have engaged and participated from 14 countries. Even just seeing this map is incredibly exciting. Just understanding, and my friend Melanie from CureMLD was sort of the influence and the inspiration behind really visualizing what it means to say we're a global organization, right? I mean, we are hearing from, I've told some of you, Saudi Arabia is participating in the data collection, Argentina, and I heard from Australia last week, they were already participating, but I heard from a family in Iraq last week. I mean, it's really incredible to just, it's one thing to say it, but it's another thing to really think about. I think you all are sharing the story of the Yaya Foundation. I think, we're getting out there. And so to see them, actively engaging in this way is incredible. We're really proud. I'm really proud of this map. I just, we get excited about different things, right? And so how did we get there? As I mentioned, Dr. Wolf joined us at a family call last year where we just talked broadly about what disease registries are and the role that they play. We've hosted a couple of info sessions since then. And I am always happy to talk one-on-one as well. So I'll just share just one snapshot as just an early insight. What's kind of fun, again, we see the de-identified data, but what's really exciting is to just see, like, for those who have participated so far, what are some of the early insights that the community is seeing at large? And this is just sort of one example of that. So as I mentioned, participation has caught the attention of our researcher and provider community. I think they're excited to see what the foundation is doing to bring families together. And there is a desire and an effort that we have committed to talking about how do we combine these these patient-reported outcomes in the data collection and map those with, provider-reported outcomes. How robust could a data set be when it's that combination? Maybe we'll talk a little bit more about, but we actually used that methodology of saying, look at the success of our data collection, look at the success so far of natural history efforts. And we submitted a pretty large grant in that vein of saying, we would aim to combine that data, and we think that that's incredibly powerful. So already that data is being used in really meaningful ways. So just real quick, like broadly, so zoom up out of 4-H and just to talk about the platform for just a second. As I mentioned, there are other rare disease groups. And so far there are 27. They constantly have like classes of another dozen that are just rolling on fairly regularly. And at this point, I think it's very safely 750 individuals who've contributed across disease groups. So again, data is swimming in a much larger pool, which is attracting a much larger, researcher group. Oh, and I should mention, and maybe it's even here, the deep dive that's happening right now within RareX is on the brain. And so they have a pretty comprehensive sort of cross-collaborative group that is focused right now on a lot of neuro, just deep diving into the neuro space so that they can roll out additional surveys really focused in our space. And then I just wanted to share, it continues on. So their pipeline is full. It's real big. So we need you. This is my explicit ask. We set a goal to try to get to 100 participants by the end of this year, which I think is a really meaningful number. Again, when we go to talk to potential industry partners and we're looking for support or funding or, just getting on their radar, really thinking about, what is that number? We think 100 is achievable and we think it's meaningful. And we would just ask that, we'd love to have you along with us. So if you've participated already, huge thank you. Absolutely. I appreciate the engagement and the time that you've taken and the sharing of the journey. If you haven't updated your password since February, this is a total technicality, but you will receive an email on Monday from RareX that's asking you to update your password. And it's because they added another layer of security for some two-factor authentication. So you will receive an email on Monday just to change your password. If you have questions about that or need help with that, I am very available to help with that. And then if you haven't enrolled, or if you just want to learn more, please feel free you'll see the URL there, and I know Shannon's dropped it in the chat. So please feel free to go ahead and visit the URL there to learn more or participate. I think that's my last slide. So let me see if anyone has questions or comments. Melanie. Yeah, so I'll repeat the question, which was specifically about RareX and, who exactly has access to it is essentially, I think, Melanie, what you're asking. So that's a great question. So Valerie and I will have access to the de-identified data after we complete a 2.5-hour pretty in-depth training that really is focused on what does it mean to have access to data, particularly in a rare disease community. The outside of the Yaya Foundation, other researchers who have also completed that training can have access to the de-identified data. The conversation that was had, we need to understand that actually a little bit more, because my understanding is that that is a possibility at some point, but it is not today. So I actually have a follow-up to do on that.
Nobody does right now. Yeah, Aaron. Yeah, so that's a great question. So Aaron was asking, is there any point in the process where the parents are asked about their data? And at this point, many of the participants are caregivers entering data. And so there are questions about like what their pregnancies were like and things like that. There is not, my understanding is a lot of questions though about the parents uploading their own genetic report, for example. But I know the genetic report upload is still like in the pipeline right now. So that's something I could do a deeper dive on as well. Yeah, it's a good question. Shannon, are there any questions in the chat? No. Okay. Okay. Okay, then I think Genevieve is online, and Nicole, we have you here in the room. So I think with that, we will transition to natural history. Does that sound good? Okay. She's online. Or she was. Let's see.
No, I'm here,
Oh, good.
but I think Nicole is starting.
Okay, I'm going to unplug these. Plug first.
Thank you so much, Christina, and especially Ron, for setting this up and bringing people together, families, doctors, researchers from all over the world. And I think it's really a very touching and also very successful story you shared with us. So thank you for making that possible. And I'm really happy to be here and to meet you in person after I've met some of you already on Zoom or via email or through MRIs and genetic information. So it's for us something we are very grateful to see and hear about your problems. So there are some disclosures now, and I think it's a good thing because it shows that things are moving. Pharma is discovering leukodystrophies, and we have some clinical trials going on, and I hope very much that that will also be the case for age, not in a too distant future. Let me share how it all began, because it began 22 years ago when I was a junior doctor in my second year of training as a pediatrician and child neurologist. And I met Sophie, who was admitted to the ward for investigations because she didn't have a diagnosis. And she was 8 years at the time. She had an MRI which looked like hypomyelination, but she was a girl, so it couldn't be Pelletier-Merzbacher disease. Moreover, she was able to walk with a retro walker, but she was able to walk. She didn't have nystagmus, and she didn't have any teeth at that time. So that was
of course, a bit of a strange combination. We didn't find any information about that combination, and she underwent a lot of procedures to find the diagnosis. She was a very cute and also very determined young lady. So she visited me many years after, when I was already in Amsterdam. So I saw her before she unfortunately passed away 2 or 3 years later. And 2 years later, I saw another girl, 4 years old. Actually, I didn't saw her. I saw her MRI on the MRI meeting. And then I heard from because I did my pediatric ICU rotation at that time. So I didn't see the child on the ward, and I asked my colleagues, how is she doing? What is it for a child? And they said, well, she walks and talks and is a very, very friendly and very happy young child. And I asked anything special with her teeth, and they said, well, she has no front teeth. So I looked at both MRIs and thought it must be the same thing. And then I started to ask around, and I think within a year, we found 2 more patients with the same combination of symptoms. And at that time, I thought it can't be so rare, because if I'm be able to find the 4 patients in 3 years' time, as someone who has otherwise not much experience in this field, there must be more patients. And in the end, it was true, and it's one of the most common hypomyelinating disorders. And look where we are now. So it's a story which shows that really cooperation is the key, because there were other doctors since who sent a lot of patients and data, so that made it possible for Geneviève and Adeline and I to have that first data collection on more than 100 patients. And I really hope you reach your number, and I will chase the European patients to get registered for your registry. So collaboration between families and doctors and researchers is really the key for those rare disorders. So in Amsterdam, we have 3 child neurologists working on leukodystrophies. Mario van der Knap founded our center. Mark Engelen sits here in the back and works on especially XALD, AMN. And my focus is hypomyelination, 4H and Pelizaeus-Merzbacher disease, but also metachromatic leukodystrophy. And that's actually a good thing, because you can really learn also from other leukodystrophies. And I will therefore compare 4H and MLD a little bit with each other. The setting in the Netherlands is, we are a very small country, so 17 million people and a very small area compared to the US. So we see more or less all Dutch patients. Children, adult patients, they visit, the frequency of visits depends on age. We also have a pediatric endocrinologist and an adult endocrinologist with experience in the disease, and also the rehabilitation physicians take care of those kids. And we are part of also a European reference network, rare neurological disorders, and there are also really efforts for treatment of specific variants, NS1 trials, and so on. So there's a lot going on, and that is, of course, also something which is very gratifying for us. So the current research we do in Amsterdam regarding 4H is on biomarkers, MRI myelin quantification, epilepsy, and the atypical forms. And we have a translational research project with Vivi Heine and Stephanie Doves and Lisa Koch, where we look at myelinating co-cultures of patient-derived iPSCs and have made cerebellar organoids. And I won't talk about that today. What makes 4H a challenge? It's a relatively new disease. So the first description of unrelated patients with this combination was in 2005. And the genes identified in 2011. And compare that with MLD. The first patient was described in 1910. Metabolic basis identified in 64 and the genetic defect found in 91. And the same is true for Pelletier-Merzbacher disease. So that's a huge extra time those disorders had. Then 4H is ultra rare, although I think it's about as rare or frequent as Pelletier-Merzbacher disease. If you look at the POLR3 related disorders, you have different disorders like the basal ganglia variant, the spastic ataxia, the very severe Wiedemann-Rautenstrauch syndrome, which are other disorders than the typical 4H leukodystrophy. MLD, metachromatic leukodystrophy, has in the beginning a normal development, and then children or adults lose things. And Pelletier-Merzbacher, we know patients with very different severities, but it's basically the same disease. And 4H has also the classical disease has a broad clinical spectrum. So what we can learn from other leukodystrophies? Just a little bit of recap. 4H is the classical form of 4H is a hypomyelinating disease, which means that the brain, which actually matures during the first 2 years, doesn't do that. So when you look at an MRI of a child with a 4H, it looks very much like the MRI of a child which has just been born, in contrast to a 4-year-old child with a normal myelination. That means there is something problematic with the white matter right from the beginning. And that's different from, for example, MLD, where you have a normal white matter, which then starts to degrade. MLD, we know 3 clinical presentations, early to late, until late adult age, actually. And as a rule, you say the earlier the onset, the more rapid the progression, and the more prominent are motor symptoms. And although, and we know also that it depends a little bit on the severity of the protein defect in metachromatic leukodystrophy. But what is actually interesting, although the disease has been known for such a long time, just I think in the last 5 years or so, the Tübingen colleagues and other groups found that there is really a group of patients with late onset who have a cognitive presentation, and they can have a stable motor function for decades. So although we know the disease for a long time, it's not that we know everything about it. And there's still a lot to discover, and that's important, this kind of information when you do clinical trials, because endpoint is often motor function. But when we know that motor function will be preserved for 10 or 20 years, that's not a good endpoint for a clinical trial. I think I can, when I think of 4H and the patients I know, and it will be very interesting whether the registry data actually support this. You can actually make I just leave out the Wiedemann-Rautenstrauch syndrome for a moment, 3 large groups, the basal ganglia disease, which is actually probably relatively frequent, I think, 4H leukodystrophy and the spastic ataxia. You can have a different kind of ages of onset and severity. In the basal ganglia disease, the very severe neonatal onset, but you also have a childhood onset, which looks a little bit different and has a much more protracted course. So you could actually relate that regarding classifications to MLD or also Krabbe disease. And the same is true for 4H leukodystrophy. And we have a group of children who do not have hypomyelination on the MRI. Sure, please. Yes, sure. I can also share the slides as PDF later on if you are interested. So. No. please do. And the spastic ataxia patients have a very specific variants in polyp3A and have a later onset. And it's, I think, important to have in mind those different kinds of presentations, and also sometimes specific genetic variants, because that might give again another entrance for treatment design. And just to show you the difference in MRI, that's a patient with a typical 4H, and you see the hypomyelination, the myelin defect, if you compare it to a normal child. 6 years later, it looks exactly the same. And that's a child with the early onset MLD, and Daphne made that picture. She sits also here, and you see that within 4 months, the white matter has been completely changed through the disease. And that makes 4H, if you look at the MRI as a biomarker, a little bit tricky. We know that the clinical presentation in different hypomyelinating leukodystrophies can be very different, although the MRIs look very much the same. That's actually a child with the 4H. At that time, that little boy was able to walk with some balance problems, but otherwise no motor issues. But when you look at the group of 4H patients, you do see that depending on the degree of myelination, the disease is more severe if you have less myelin. And also when you get older, you lose brain tissue, especially white matter, and that's also correlates with increase in severity and disease severity. We look at quantifying myelin with MRI, and we look at different technical parameters. And what's quite striking, although you see such a clear difference between hypomyelination and normal MRI, when we look at those quantitative MRI parameters, 4H or hypomyelinating disorders are the most normal of all leukodystrophies. And that's also something we need to understand if we want to design clinical trials. And I would like to show you a spectroscopy where we measure certain chemicals in the brain, and you see that looks very much like a normal brain. And when you compare that to MLD, for example, you see that that looks really different. For example, this NAA peak has been completely lost. Body fluid biomarkers, we don't have good body fluid biomarkers, whereas we do have them in MLD. We looked at a neurofilament light chain. That's a protein which is released by the nerve fibers when they are damaged. It's not disease specific, so also after trauma you can get a neurofilament light chain in your blood and in your CSF. But you also have to realize that if many neurons are already gone, your NFL gets low again. So in very severely affected patients, it can be that the NFL is quite normal when they are very stable at that stage. But Shanice Verepot, another PhD student, showed for MLD that actually, a bit contrary to our first expectations when we looked at only a few patients, it's a useful biomarker, especially in the young patients, and especially just before the deterioration starts. And when we looked at 4H, I was at first a little bit disappointed because it was relatively normal. But when you look closer, you do see that it is elevated compared to controls. I didn't do the exact analysis, but just from knowing the patients, I did that test. It's more elevated in patients with more severe presentation. So I think that this is a potential biomarker. I shared those data with Adeline van der Ver for the grants. So I hope we will see more about that. So I think we can learn a lot from other leukodystrophies, and also learn a lot from the fact that we really got into detail, for example, in MLD with those very circumscribed special presentations quite late. We should do it really well now. And we need also good biomarkers because that's all important for future clinical trials. We need a good and meaningful outcome measures, but it shouldn't be too complicated to collect all those data, because if you collect a huge amount of scales and scores, I'm not so sure whether you would go for years with your child 4 times a year to a very extensive testing battery. So we need to look for good scores, which are also robust, which you might also do remotely, certainly in times of a pandemic. And I hope that we will all together manage that in the next years. So thank you all for helping us with this work, and also all the doctors who sent a lot of scans and patient information and skin biopsies and so on. So thank you, and please ask your questions. Okay, thank you. The question is, what's the difference between hypomyelinating and demyelinating leukodystrophy? In hypomyelination, you don't make myelin, and that's from the beginning. So from birth, so myelin is never properly formed. In demyelinating leukodystrophy, you have a normal myelin development until a certain stage when the disease starts, and then the myelin which is there is degraded quite rapidly. So that's what we call demyelination. Sure. Oh, thank you. So the question is whether the myelin the body does make, or the brain does make, whether that's of a good quality. We don't know. And that's something we would like to find out with those extra new MRI techniques. And that's also something I hope we could explain the difference between some patients, although the MRIs look so very much alike. So perhaps that helps us. Yeah, thank you. So the question is whether when 2 patients have the same MRI, whether they present similar. When I see a patient, no, actually no, I have to say that the short answer is no. There is a longer answer, but the short answer is no. When I see an MRI with hypomyelination, I never know whether the patient is walking into my room or is coming into a wheelchair. And has also other severe motor problems like with hand function. So I can't see that well on a simple MRI. And that's a question which we've asked ourselves already for a long time, and we still didn't come up with a good answer. Honestly. Yeah. 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 Yes, we have also severe polar 3D cases. As a group, the polar 3D cases are milder, but within the group of polar 3D cases, we also have severe ones. Yeah, that's one thing we are working on, and also I think your grant application is about. It's an important question, and we all would like to have more measures to better predict disease course. Oh yes, excuse me, sorry. So the question was that whether there are good measures to predict deterioration of patients and the severity. No, there are no good measures, but many people are working on finding more information and more informative measures for that question. Yeah.
I don't think I've ever seen like the data before about NFL.
Mm-hmm.
Could you share a bit about the usefulness of it?
Mm-hmm.
Biomarker and the big usefulness of it?
Yeah, yeah. I think that those data are actually at the moment not enough to understand the use of the potential use of NFL. I think we need more. We need more data points, and we need a good correlation with the severity and the stage of the disease. But I think when I look at that, and when I remember our experience with MLD, where I was also very skeptical at the beginning that it might be useful. The NFL gives us information about the nerve fiber and the nerve cell, but we would also like to have a biomarker for myelin, for example. So how much myelin, how good is the myelin? And NFL is a very indirect biomarker. So I hope we will also find at some point a direct biomarker for myelin.
Maybe the whole increase is a little bit lower. So I'm wondering, have you looked at longitudinal data?
Yeah. So the question was about the NR spectroscopy, where we measure those chemical substances in the brain, certain chemical substances in the brain, actually, we have a lot of longitudinal data on 4H and other hypomyelinating disorders, and I would love to look at them, but I need someone to do that. And that's still actually sleeping in our data collection. Yeah. So what we hypothesized was whether the NAA is very slowly decreasing. One of the problems why we just can't pull out data and look at them, and after 2 weeks we have the answer, is that when you look at a very long time frame, you have usually your hospital buys another and perhaps another MRI scan, and you always need to calibrate those data and to find a factor to compare them well. And that sounds a bit basic, but that's how life is. Huh? You never can do the same, use the same MRI for a period of 10 or 20 years, because there will be better MRI scans, and so that's one of the really daily life issues we deal with. So the question was about the adult onset, and that's quite variable. We see patients with balance problems or late onset tremor, or also mild movement disorder, but we also see patients with a psychiatric presentation, like a psychosis, or very prominent behavioral problems. And almost without, or only very mild motor issues. Very behavioral, yeah. Yeah. So the question is about remyelination, whether there are studies on remyelination, not for hypomyelinating disorders yet. There will be some, but not yet, and not for age at the moment. I'm afraid. Mhm. Yes, you're right. Yes, yes, yes. So the question was about volume loss. You are completely right that also normal people have a volume loss of the brain that sets in at around age 30. But for age, it's much earlier and more common. So you do see the difference. Mhm.
We're not the same, I guess, or
Mm.
sub 4 and 4-3 are sort of tied together and you mentioned sort of separately.
Yeah, thank you. So the question is about the different disorders or presentations, and you're right, I should have explained it better. We when we started with what is for age, we came from those children who had this MRI scan with a lack of myelin and also a loss of volume of the cerebellum, the small brain. So that was what we knew, and the combination with the teeth, and in the older children, the puberty delay. So that was what we knew as 4H syndrome. Then the gene was found, or the genes leading to 4H syndrome. And then we found more patients with more severe, milder disease. But then nowadays we look differently with genetic testing. So at that time, when we found the disease, so the disease gene in 2011, we did often, we saw that patient, we thought it could be 4H. We looked at the polyR3 genes, we found the variants, and we had another patient. Nowadays, you often do it the other way around. You have a patient with a neurological disease, you don't know what it is. You do a very broad gene testing. It's called whole exome sequencing or panel sequencing, where you look at a lot of genes at the same time, and then you can find variants in a gene you didn't expect. And that was the case in patients who had actually almost normal myelin, but very small central parts of the brain. And that, I think no one of us would have predicted that this would also be related to polyR3A. But that's that disease group. Yeah. So the question is about hearing loss in 4H. To be honest, I don't have so much experience on hearing loss in 4H. I've seen a few patients with that problem, but it's not the rule. So I think that's a question, a very good question also for the registry. So we hope to find more patients with it. I think, should we move on?
So Genevieve, I think if you're able, it sounds like you have some slides as well. So I don't know if you're able to share your screen.
Yes. Can you see it?
Yes, we can.
You see the presenter mode?
Yes, we can. Thank you.
Great.
All set.
Excellent. Okay. So hi, everyone. I'm sorry, I can't see you. Can you hear me well? Okay. I assume so. I can't hear you either now. Okay, I wish I would have been able to be there in person. I really missed coming to the ULF and seeing all of you. I want to first thank Randali and Christina for inviting me to talk about the natural history study. Nicole and I had decided to talk about slightly different things. So I'm going to focus on what we're actually collecting right now. So none of my disclosure have anything to do with what I'm going to talk about today. I've been involved in the clinical trials, and I'm also involved in different foundations, and I'm also working as an editor on some journals as well. So you already heard some of this. So what are biomarkers and surrogate markers? There are things that we can look at clinically to help us understand disease progression. They can be MRI features, some of which Nicole talked about earlier, and also measuring some specific chemical in the body can either in the urine or in the blood, and they are very important because these help us follow the disease and response to therapy. So the natural history study basically is a retrospective and a prospective natural history study. Retrospective being looking in the past, and prospective is actually following the patients over time. And we also have other related studies that we do to try to better understand the disease as a whole and the different characteristics of the disease. So we look at the normal developmental milestones for patients with 4H. We look at the craniofacial characteristics, the dental abnormality. We look at bone health. And then we also have studies about looking at how the patients and caregivers live with the disease and how we can help them better. So the retrospective natural history study and the prospective natural history are done very closely in collaboration with Nicole Wolf and Alan van der Veer. And in my lab, it's done by Hilja, who's soon to be a PhD student. And we are working very closely with the Yaya Foundation. So in the retrospective natural history, we ask patients to send their charts, their medical records that we review. We ask about clinical acquired MRI, and we also do the questionnaires that we're going to talk a little bit about later. And in a prospective natural history study, we look at, we basically bring the patients in or do specific assessments remotely. We do blood and urine tests. We can do research MRI, and we also do questionnaires over time. For now, the prospective natural history study is on hold because of pandemic and also because of the lack of funds. So the database we're using for the natural history study is called LORIS. It's a database that was done at McGill, and that is an excellent database for longitudinal data collection, so data collection over time. And we are working, the grant that we submitted recently would hopefully cover for the two databases, the RareX and the LORIS database, to be able to talk to each other. And then the related studies. So we look at the developmental trajectories of patients with 4H, and that's going to help us understand how they acquire milestones, developmental milestones, and then better understand and stratify patients for clinical trials. And for this, we sent you questionnaires to fill out. And for those who already did, thank you so much. Aline and Elia are both responsible in my lab. As you may know, Polar1C, which is one of the gene causing 4H, also causes a disorder that's called Treacher-Collins syndrome, which is a disorder with some craniofacial, so some facial characteristics that are specific to this disorder. So we're working on a project where we look at patients with Polar1C but other mutations and other genes and look at their facial characteristics to try to understand if there are specificities to the facial characteristic of the patients. And that's done by ANITIS. We ask you to send medical dental records for your for your children, as well as the x-rays of the teeth. And we also, when you come to the children, do specific pictures of the teeth, again, to better understand this specific manifestation of the disease. So bone health is a very important thing to look at, because it can lead to, if it's not been taken care of properly, it can lead to fractures and pain. So it is very important in our patient population to look into it and see whether or how we can help better. Patients with 4H are at risk for osteoporosis because of their underlying disease, because of the motor difficulties, especially when they are dependent on their wheelchair to move around, but also they also at risk because of the endocrine manifestations that are associated with the disease. So for this, we send you questionnaires again. Thank you again for the ones who have taken the time to fill them. We also have medical records relating to bone health. And for those who come to the children, we do x-rays, blood and urine tests. So we send you other questionnaires to look at how the parents and the patients live with their disease. So we look at the quality of life, and we also look at parental stress. And this project is actually to look at factors that we can modify over time to improve the patient's and the parents' journey. But these questionnaires are very focused on specific aspects, and they don't cover everything that the parents may want to say to us. So we also have just recently started work for patients and families, and that will be done with Adam, who just joined the lab. And this is again to identify modifiable factors that we can work on to improve the patient's and the parents' life over time throughout the disease course, and to be complementary to our questionnaires as well. So you may get some in the email about whether or not you're willing to have an interview with Adam or someone else from my lab, or a phone call. So it was a very short talk about just to have you informed about what we're doing right now in the lab about natural history studies. So I wanted to first acknowledge patients and families without whom we wouldn't be able to do what we're doing. I'm not going to introduce everyone on that slide because there's many people. I'm going to just tell you that Alan Evans is the investigators that did LORIS. So his lab and my lab have been working very closely to adapt LORIS for the purpose of the clinical manifestations and the disease course for 4H patients. And international collaborators. So I wanted to highlight Adeline and Nicole, with whom we do the natural history study. But also Gwen Pingao, you'll hear about some of the work that we've started doing on preclinical work on gene therapy for 4H. These are my research staff. So I have 2 research associates working in the wet lab. I have 3 clinical research assistants. And these are my graduate students and medical students or pediatric neurology residents working on different projects. And I'm very grateful for the funds that we've had over the years for Project on 4H, but on other leukodystrophies as well. And I want to highlight the Yaya Foundation for their support for the mouse project and the gene therapy project, and for our very nice collaboration over the years. And I also remember very well when we were sitting in the room and talked about how a foundation would be a tremendous help for 4-H leukodystrophy research. Thank you. I'm happy to take any questions.
Does anyone have questions for Genevieve? Okay, Tammy, go ahead. How well can you hear Genevieve? Maybe not at all.
I can't hear at all. No, sorry.
Okay, so you know what? She'd probably rather hear from you. Oh, go for it.
If you come to the front, I'll also be able to see you, and that would be really, really, really nice.
Oh, sure. Okay. Oh, sure. Okay.
Yeah. Oh, you don't have to, but I haven't seen you in so long. Hi.
Hi, Dr. Bernard.
Nice to see you.
You too. Thank you. My question was, when you see patients in Montreal, 4-H patients, you mentioned doing urine and blood tests. What are you looking for when you have those tests done?
So we, so you saw Nicole Stock talking about neurofilaments. So these are blood tests that we isolate plasma and serum, which are components of the blood. We aliquot the urine, and then we've done that over the years for several patients that we've seen in Montreal. And the hope is that we will be able to go back in time and look at a specific biomarker and then correlate this with what we have in the Loris database on disease progression. So we would hope that a good biomarker, for example, neurofilament or something else, would change over time as the patient is changing. So that when we do a clinical trial, if we want to follow this specific biomarker, we will know if the biomarker goes up as the disease progresses. We would see it go up as the disease progresses, and then we would start the potential therapy, and it would go down again. So we've been banking these samples for almost 10 years now, but we haven't looked specifically for biomarker yet.
Okay, so my question then, because we're in the US, is there a way to get them to you in Montreal, or should we come to see you so that we can also bank those samples?
So I know Dr. Van der Veer. I don't know if you're going to her lab, to CHOP as well, but I know Dr. Van der Veer also is banking samples, and Dr. Wolf as well. You're welcome to come to Montreal. We would love to see you there, but I wouldn't want you to come just to send blood and urine tests. We can arrange blood and urine tests and have them sent to us. So Tik Luan is probably in the room. He knows all about how to do this. If you live close to CHOP, it may be easier to go to CHOP. But if you're further away, we can easily organize some blood tests and urine tests and then have them sent to us.
Okay, thank you. Sorry, one more question. I'm sorry if Dr. Wolf has already mentioned this. How close are you then to being able to use the blood and urine samples of 4-H patients as biomarkers?
So my lab has no funds to do this. So we haven't, we're just banking them. So we haven't worked with them yet. But like if, I know that we have 2 potential candidates for biomarkers. They're very, very preliminary. One is the neurofilament. The other one would be like a small RNA. So as you know, PAL2, which is the 17 subunit complex, transcribe DNA into RNA. So it is possible that some RNAs are can be measured in the blood and then can be biomarkers for disease. So we do have, that's one of the reasons why we're banking these samples, but we haven't had a chance to look at them because of lack of funding. But if we ever have funding, then we'll have the samples. It's much easier to have the samples than to start collecting the samples when we have funds, because then we're 10 years behind.
Thank you very much.
You're welcome. I'm happy to take any other questions. You don't have to come to the front. It's just nice to see you guys.
Hi, everyone. I just wanted to let you guys know that my name is Luan. I do work on Dr. Bernard's research team. I'm a part of a research staff, and I would love to connect with you guys maybe during the afternoon or at a break or something, and we can talk more about the research. And I have a bunch of consent forms. If you're interested in participating, I can talk about that as well, and we can get everything signed and make sure everyone's set up. Okay, thanks.
Thanks, Juan.
Hi, Dr. Bernard. Mason Tallman's mom. My name's Rebecca. How are you?
Hi, nice to see you.
Nice to see you. How do we get our children to CHOP? How do we get them seen? How do we make that happen? To start collecting urine and blood and have the specialist eyes on them. Mason has not been seen by a leukodystrophy specialist in probably 10 years.
And you're close to CHOP?
We're in Chapel Hill, North Carolina, but willing to travel. I just don't know who to talk to make an appointment.
Okay, I'm very bad in geography, so you could come to us or you could go to CHOP. I think Luan can probably put you in touch. If you're closer to CHOP than to Montreal, then Luan can put you in touch with Omar, which is Dr. Vanderveer's Luan.
Okay.
And then I'm sure Omar would be able to organize a visit, but I'm not sure exactly how they work. As you may know, the healthcare system is very different in the US and in Canada. So I'm not exactly sure how it works, but I'm sure Omar would be able to help.
Thank you.
You're welcome.
Genevieve, there are no questions. I should look in the camera. There are no
There's-
questions in the chat.
There is one
So-
in the chat. Sorry, that just came from Georgia Youth. Oh no, sorry.
Okay.
Wait, it
So,
was a private question. I'm sorry. I'll answer that in the chat.
okay. Well, Nicole, I'm sorry. I don't know if I thanked you appropriately. So thank you so much. And do you have, Kathy, you have a question? Do you want to come up front? Or I can ask it for you too. That's okay. Sorry, I'm so sorry.
Dr. Bernard,
Yes.
hi. You were saying that you were collecting samples, but you didn't have enough money, so you're basically banking them. How much does it cost to go to the next step?
So it depends what the next step is. The next step is to generate preliminary data. I think both Nicole and Dr. Vander and Adeline are doing that right now. I don't think they have funds either. So I think we've been working together to not duplicate the work and to make sure that we can get preliminary data for grant proposals. It's a complicated question because in order to look at your biomarkers and look into measuring biomarkers, you also have to have clinical data at the same time as you do the biomarkers. So you have to have a way to look at what's the stage of the disease at the time the bloods are taken. So it's not just money for this that we need money for a natural history study. And we've been working very closely with the Yaya Foundation to find solution to try to have those funds. But these are very expensive, especially the prospective one. So the one where you see the patient regularly in clinic, or you do remote assessments. The retrospective studies are a little less costly. They are costly, but they're less costly. And the way we've, like, even if we do retrospective studies, we have some patients that we had samples on over the years. So we'll be able to look at these potential biomarkers in a retrospective fashion. I'm sorry, it's not a good answer.
No,
to your question.
Dr. Bernard, it's Ron. Can I try to tag on to that? It was a great answer because it's a hard question. The dollar amounts are really high. And it's something that, so I referred to it really quickly in my intro, and it's something that we as patients can do. Like, for example, we submitted a joint grant to the Chan Zuckerberg Initiative, another one, for $2 million to do prospective natural history work and biomarker identification. And even that won't cover all of the work we want to do. And so the foundation would sort of provide some supplemental funding on top of that $2 million but there's a lot of work to be done and lots of ways that we can go about supporting it together. And that could be fundraising ourselves, sharing our story, writing letters to the NIH. And so we're working right now on sort of a plan A, B, and C approach to try to get this work that Dr. Bernard and Dr. Wolf and also Adeline are talking about funded. And we would love to talk to you more about that and our sort of collective strategy and how we can go about working together to try to do it. So. Well, let's talk. Yeah, thank you.
There are other questions? Shannon, anything in the chat? Okay. Well, again, Dr. Bernard, Dr. Wolf, incredible, a huge amount of thanks, not only for the presentation and the Q&A, but also more importantly, your work and your care for these families and the Yaya Foundation. So thank you so much, both of you.
Thank you for having us.
Great. Well, it's really a pleasure to be here. I'm a first timer at ULF, and so I want to thank the ULF for making this meeting happen and bringing us all together, scientists, families, patients. It's really a unique meeting from that perspective, and one that I'm certainly taking away a lot of information and interactions from that I think are really valuable. I also want to thank the Yaya Foundation. It's appreciate being invited to present our work here, and it's been great to meet everyone in the leadership of the foundation in person after having interacted online a few times. So the topic of my talk today is about making mouse models of RNA polymerase III-related leukodystrophy or 4H disease. And so I'll just get started here. So, this field, as we've heard already this morning, it really sort of took off in 2011 with a series of landmark papers in the American Journal of Human Genetics from the Bernard and Brace groups at McGill University and their collaborators in the Matsumoto group in Japan that established the genetic identity of the mutations that unify this disparate group of diseases that have these variable phenotypes that we've heard about so far. So these are landmark studies because once we knew genetically where to look, it was possible to identify the genetic defects in people that we united by a common set of disorders and phenotypes. So this led to additional important advances in the clinical spectrum of the disease, as we've already heard from Nicole about this morning. And this has led to a growing number, continuing to this day, mutations in the RNA polymerase enzyme that are associated with these various phenotypes, and the remarkable phenotypic variability that we see between patients with different mutations, or even sometimes with the same mutations, that suggest genetic modifiers in our human population that modify disease severity and other kinds of phenomena that can impact how the disease is manifest in the patients. So in really what's been a relatively short period of time, only 11 years since the genetic identification of this disease, tremendous amount has been accomplished. And a part of that has been, the development of important research tools to study the cellular and molecular basis of poultry-related leukodystrophy. And this includes cell lines engineered to contain mutations found in the patient population, patient-derived fibroblasts, and as we've heard, iPSCs that can be turned into oligodendrocytes and other cell populations that express the mutation. And so this has been really informative in getting some initial insights into molecularly what's going wrong in the cells that have these mutations. But there's been one area, the generation of animal models, which has been particularly difficult to make progress on. And so it's been a challenge to make these models, but these models are really critical for understanding the disease in a context where all of the cells are there together in a mammalian model. And that model can be used ultimately to understand disease pathogenesis and to test potential therapies before they go into clinical trial. And so it's our efforts to try and do this, which I'm going to be talking about today. But before I get into the details of how we've been going about making animal models here, I think it's helpful for you to have some perspective about the field in which I've been working, and many others as well. And so the field is of the polymerase III transcription is really quite advanced, and so it's a mature field of research. It's actually been going on for over 40 years, which is about the same amount of time that the ULF Foundation has been meeting, right? And so the fundamental knowledge about RNA polymerase III transcription is really well advanced because of the many years and decades that people have been studying this system. And because of that, this is what we're hoping to try and leverage to really make a rapid progress in understanding the molecular and cellular basis of the disease and help to inform ways to develop new approaches to therapeutics. Oops. So a vast amount of information has been obtained over this period using model organisms from yeast to human cells, frogs, flies, worms, everything you can imagine, as basic systems to try and study the biology of polymerase III transcription and understand its impact when it's perturbed. The machinery of the poultry system is really well understood. People have been able to take extracts from eukaryotic cells, add exogenous genes to those extracts and have them transcribe RNA polymerase III transcripts, make them in vitro, and by that very nature to fractionate those extracts and understand the nature of the proteins that are required for this process. And so this is all very well known and described these days. In the case of the most commonly studied type of RNA polymerase III transcript, the tRNA genes, these are the most abundant RNAs in eukaryotic cells, or any cells for that matter. And their transcription requires just 26 proteins. There's 6 proteins that are made up by a single transcription factor that binds to the promoters of these genes. That transcription factor recruits a 3 subunit initiation complex to the genes, and that assembly is then recognized by the 17 subunit polymerase, which is carrying out the transcription. So it's a relatively simple system in terms of the number of components. The regulation of the system has also been studied in great detail under diverse conditions. It's a system that's extremely sensitive to nutrients and nutrient stress, and in fact, all kinds of stress, environmental stresses and cellular stresses. It's also a system that is targeted typically in oncogenic transformation in cancer. Pol3 transcription is vital to cell growth, and under conditions where cell growth control is lost, this process is upregulated, and so the Pol3 system has been heavily focused on to understand growth dysregulation in cancer. And as a consequence of that, we know a lot about the major signaling pathways and key regulators of this process. The function of Pol3 transcripts themselves are generally well described. I've already mentioned the tRNA molecules as the major component of that. These molecules, of course, we refer to them as the interpreters of the genetic code, because they carry the amino acids to the ribosome and recognize the triplet code in protein synthesis. There are many other types of Pol3 transcripts that are made in our cells. They have very diverse functions, from roles in messenger RNA splicing, protein secretion, regulation of gene expression, and others. And so we know quite a lot about these functions. But RNA is a bewildering molecule in many ways, and we're still learning about new functions or non-canonical functions for RNAs that we've been studying for many years. And one class of these RNAs, which is relatively newly described and his studies are still ongoing, are a class called tRNA-derived fragments, which are pieces of tRNA molecules that are generated by their partial breakdown that actually have other biological functions. And all of these things, these TRFs, the normal functions of tRNAs, and the other Pol3 transcripts, all of these can perturb the biology of our cells in ways that could potentially impact disease. And this is what we're interested in trying to understand. So overall, there's a wealth of information to consider in the context of disease, whether that disease is leukodystrophy or cancer or immune disorders and so forth, and so on. And as I've already said, this is what we're trying to leverage in the context of 4-H to move things forward. So, my lab has been doing this research at Albert Einstein College of Medicine in New York for some 34 years. I started as a postdoc before that, working in this system, so I have a long history working here. But the field is quite large and active, and I have an image to show you of a group from a recent meeting. So we get together every 2 years and have done so since 1986 to present and discuss our work on this system. We did this as an isolated Pol3 community in the early days, and in the late 90s, we gathered together with the people working on RNA polymerase 1, realizing that there are many commonalities in the systems that we study. RNA polymerase 1 makes the large RNA components of the ribosome that's involved in protein synthesis that Pol3 transcription also participates in. And so we've been meeting together for some 12 meetings now. And given the names of our enzymes are, numbered oddly, we're affectionately labeled ourselves as the Odd Pols meeting. And in recent years, we've been attracting other groups who may not have odd names, but are nonetheless odd polymerases. And so we welcome them to the family as well. Okay, so let's talk more about the models and how we make them. So modeling Pol3-related leukodystrophy. So this, as I said in in my beginning remarks has been a challenging problem. And the challenge here is in part because while mice are indispensable for modeling human disease, they're not human, right? And conversely, humans are not mice. And so this sometimes comes home to roost in your experiments where you find that something that you expect to have occurred using a mouse model for a human disease, in fact, doesn't happen. And that was the case in the first mouse model of poultry leukodystrophy that was generated by the Bernard and Brace groups at McGill in work that we were happy to be involved in a small way. But the experiments involved generating a mouse that contained a homozygous polar 3A mutation, a founder mutation in the human population, but it didn't have disease. It was perfectly fine. And moreover, poultry transcription in those mice appeared to be normal. And so this sort of suggested that, in fact, mice are less sensitive to the diseases, to the mutations that cause disease in humans. And in fact, what we needed to do was to make a stronger mutation in order to model the disease in mice. So this is what we wanted to do. And the approach that my lab took to do this involved using budding yeast, my favorite small organism, because you can make beer and bread with it, and I like brewing, right? To use budding yeast as a eukaryotic model system to try and stratify the effects or rank the effects of severity of different mutations to make a stronger allele. And so we generated a series of single and double mutant disease-causing poultry leukodystrophy mutations and put them into yeast and measured the ability of the yeast cells to grow on agar plates. And that's what you're looking at here. So yeast's favorite temperature is 30 degrees. That's its optimum. And what you can see in this panel here is that here's a wild type strain and a series of single mutants. And down here is another single mutant. This is the founder mutation. And when you look at the relative growth of these strains on this agar plate, they're all the same, right? So there's no phenotype for these disease-causing alleles in yeast, similar to the founder mutation in mouse, in fact. Right? So we've made a number of double mutants combining different disease-causing alleles. And fortunately, some of those had some phenotypes. And you can see two examples here, one in the red box, where growth at 30 degrees is diminished somewhat, and clearly more severely in this other mutation. And that's at the optimal temperature. When we shift to suboptimal or non-optimal temperatures, we see heat-sensitive and cold-sensitive phenotypes. So these differences in the strength of the growth of the cells on this plate is an indication that enzyme function has been compromised and the ability of the cells to survive and grow has been altered as well. So given the properties of this particular mutant in growth in this particular experiment, we did a number of other experiments on that particular mutant and found decreased expression of poultry transcription in yeast cells. This is good. Decreased transcription by purified RNA polymerase 3 from those cells in the test tube. So we knew the enzyme had a measurable biochemical defect. And this became the mutant then that we chose to engineer into mice to try and model the disease. So we made the construct in the mouse, and we certainly got a more severe disease phenotype. Unfortunately, we overdid it a little bit, right? So the mice that are homozygous for this double mutant polar 3A mutation, they don't survive embryogenesis. So this is not a viable animal model. So at that point, we had to retool our genetic approach. And instead of expressing this mutant gene in all cells in the mouse, just pick on a population of cells and target them and see if we can generate viable models that have phenotypes. And that's what we're able to do. we used conditional expression of this mutant in order to get viable mice to study. And in choosing how we would do this, we were motivated by the hypomyelination phenotype that's seen in the MRI images of patients. And so we targeted, as a result, cells of the OLIG2 lineage. So these are the cells that comprise primarily the oligodendrocyte precursors, and the mature oligodendrocytes are the cells that make the actual myelin. And it was those cells, and only those cells, that were expressing the mutation. And we were able to generate viable animal that exhibit a subset of clinical features that are seen in patients. So I'm going to show you some of those phenotypes in the mouse now and try and relate that to the patient. So what we see in these mice, we call them POLR3A conditional knock-in or CKI mice. They have impaired growth as neonates, as you can see here from birth to 21 days of age, which is when the mice are weaned. In the males, quite pronounced difference in body weight, which persists in the animals as they age through adolescence and adulthood. You can also see in older animals, they're not as long. Their size is definitely smaller. And so this growth defect is something that, or growth difference is something that is sometimes seen in the patient population. In testing these neonates during this very early period, we also noticed that they had delayed acquisition of developmental milestones. And that was the first indication that we maybe had some neuronal defects in these animals. We've looked at this extensively in the adult population and identified, using behavioral studies, defects in cognitive function, thermal and acoustic sensory responses, and fine sensory motor control, all of which is suggestive of a hypomyelination defect, resulting in the decreased nerve impulse conductance. So, our original goal when we set out to do this was to try and develop a hypomyelination model. So did we manage to achieve that? The answer is yes. And so our mice do in fact exhibit hypomyelination in discrete brain regions. So here are just two examples of several experiments of different types which demonstrate that point. Over here, we're looking at immunofluorescence of myelin basic protein, one of the constituents of myelin. And what you can see in the cerebral cortex, here in the wild type versus the knock-in, or in the corpus callosum, here in the wild type versus knock-in, or in the hippocampus, wild type versus knock-in, a reduced intensity of staining of this myelin basic protein, indicating the hypomyelination phenotype. But it's not throughout the brain. And in fact, it's quite striking that we don't see any changes in the staining of this myelin protein in the cerebellum. This is a result we still don't understand, but it's an intriguing observation that the phenotypes are not fully penetrant across all brain regions that contain myelin. So here's a different example of, that shows a slightly additional features of this hypomyelination phenotype, transmission electron micrographs of the corpus callosum. So here again is the corpus callosum. This is a bundle of nerve fibers that connect the right and left hemispheres of our brain. And when you cut that corpus callosum perpendicular to the plane that you're looking at and turn it on its end, you're looking at a cross section of the nerves. And you can see them here in the wild type, very prominently displayed with this sort of outer ring that's very clear. And when you look in the knock-in mutant, where have all the myelinated neurons gone, right? They're certainly underrepresented here in these micrographs. The neurons are actually there. You can see them at higher magnification much more easily. This is only 5,000-fold magnified. So the neurons are actually there, but they're not myelinated anymore. And so when you count these, you find that there's an increased number of unmyelinated axons in the corpus callosum of these knock-in mice. that's not the only characteristic that you can uncover in looking at these images. When you look at the myelinated neurons, and you can measure the thickness of the myelin layer, which is the black layer that is ensheathing these neurons. You do this on hundreds of neurons. You find that the myelin layer is not as thick, right? And so it has decreased insulative properties in the knock-in relative to wild-type animals, and presumably this contributes to its diminished capacity as well. So there are really two major findings that have come out so far of the studies on this particular model, and they're indicated here. That the disease pathogenesis involves defects that reduce both the number of mature myelinating oligodendrocytes and the ability of those cells to produce a myelin sheath of normal thickness. And I haven't shown you all of the data to support that point, but you know, we have it, and it's in fact published in this article that we reported last year. So we're continuing to work on these animals to learn more about what's happening at the cellular and molecular level. But we wanted at the same time to expand our view of what this particular mutation could do to cells other than the oligodendrocytes. And so we've made a new model to do this. I'm going to present just one slide showing this model and what we've learned about it. This is unpublished work. And this particular model, a second model of Pol3-related leukodystrophy, is one we call POLR3A TAMKI. And this is a whole body inducible Pol3-related leukodystrophy model. So the way that we have engineered this mice is that we're using genetic tools, very familiar genetic tools, where the mutation can be conditionally induced in all cells in the mouse, but we do this postnatally. So the animals they don't have the mutations from conception. They only develop disease after we treat them with a small molecule that induces the recombination that leads to the expression of the mutation. And so what's important about this model from our perspective is that it allows the assessment of the effect of the leukodystrophy mutation on the entire CNS population, not just the oligodendrocytes, but the neurons, the astrocytes, the microglia, and so on. But it also allows us to look at potential effects of the mutation on non-CNS tissues, right? And so this, we hope, will help inform perhaps some of the other phenotypes we've heard about at this meeting. And so our experimental regimen here involves treating the mice with tamoxifen about 7 days after they've been weaned. We do 5 treatments every other day. And then in the first series of behavioral experiments that we've done, about 14 days after the initial injection of tamoxifen, we put the animals in a behavioral spectrometer. And this particular instrument has a video tracking that allows us to monitor the mice's activity in this chamber over a period of time. And then pattern behavior recognition software can go in and look at what the mice are doing and identify particular behaviors that have been impacted. And what you can see down here are some of those behaviors that relate to locomotion, right? So this is the track length. So this is measuring the distance that the mice are covering in the chamber over a particular period of time, either 6 or 9 minutes. And you can see pretty clearly that this whole body knock-in has reduced track length relative to the wild type animals. They're not moving around the environment to the same extent as wild type. Right, they also spend less time running, and they also spend more time standing still, right? And these are the behaviors shown in the center and the right. And those behaviors seem to get worse as a function of time. So as we look at, say, 42, 49, 56, and 63, you see the bigger difference or increase in effect in the knockout relative to the wild type. And so, so this piece of information and other data that we have suggests that that these mice have a more severe course than the original Pol3 CKA mice, where the mutation was just limited to the oligodendrocytes. And we're hopeful that this is going to provide much more information about the cellular impact of the leukodystrophy mutation that we can learn from and help to inform what's going on in patients. So I think that's really all I wanted to present today, to tell you sort of an update of where we are and where we're headed. So the key points, if you take nothing away from what I said, this is your slide. We have two models in mice now that reproduce phenotypes of Pol3-related leukodystrophy. And these enable studies on the cellular and molecular basis of this disease that will lead to a better understanding of disease pathogenesis. And importantly, the mice will provide a critical model for preclinical genetic and pharmacological testing of approaches that we hope will be effective at mitigating the consequences of this disease in patients. That's our goal. So I have some people I need to acknowledge, the people in my lab who have contributed to this work. A graduate student, Emilio Mehab, who will be presenting his thesis defense in the next couple of weeks, has been one of the important drivers of this work. Christian Levidos and Robin Moyer, who is not only you know, the right hand in the lab for me, but a huge source of academic inspiration and in fact a co-PI on our grant support for the National Institutes of Health. We've also had great support from people in the Department of Pathology in the analysis of electron microscopic images and immunofluorescence studies. Maria Gallinello in the Animal Behavioral Core, obviously we've depended a lot on her, and she's been fantastic at training Emilio in these kinds of experiments. And in other studies that I haven't shown you, we've also done magnetic resonance imaging in these animals to try and use a technology that is so important in the patient population, where we can see similar kinds of things going on. And that data is also in the paper that we presented last year, but I haven't shared it with you today. But the people in the Grus Magnetic Resonance Center were vital in collecting and analyzing those images, of course, too. So that's all I have. Happy to answer any questions if you have any. Yes.
First of all, congratulations on the beautiful house.
So the short answer is we haven't really gotten to those experiments yet. We definitely want to. Yeah, sorry. The question is, have we begun to analyze these mice in terms of their hypomyelination phenotype, or looked at the development of this phenotype as a function of time to know when should we start inducing the disease in this model? And so the answer is no, we haven't really gotten there yet. We decided on the particular regimen that we used just out of convenience, right? So the mice have been weaned for 1 week. That's when we start the treatment. So they have a little bit of time to get used to not being with the mother. And then we induce the disease. It certainly is possible in this model to induce the disease earlier. One can deliver tamoxifen to pregnant dams to induce recombination in the pups in utero. That comes with some risk of the pups not all making it to term sometimes. So we may be limited to some extent in doing it that early. But clearly we can push things longer as well, and it might be interesting to do that and see sort of maybe a late onset type of leukodystrophy model using this particular system. Yeah, thanks for your question. Yes. It's very difficult. So the question is, how does the phenotype of the yeast strains correlate with the patient population? And it's really difficult to draw straight lines between these things, in part because of the variability of phenotypes in the patients, right? I think things are a little more straightforward in yeast, and we can certainly rank order the severity of these mutations quite readily. That's not to say that yeast doesn't have genetic modifiers of phenotype as well. They most certainly do, but we haven't been able to connect the dots in that way. Sorry. Yes. Yes. So, I mean, things were definitely slowed down by the fact that the mice, sorry, the question, the question is about the time frame for development of the knowledge from sort of start to finish with respect to this animal model and how that compares to other disease models. It could have gone a little faster if we hadn't have had the problem with the mice not exhibiting the same sensitivity to the mutations as the humans apparently do. So that could have trimmed maybe 2 years off the process. So for us, we got started in this probably in 2016. And, you know, we had enough data by the end of 2020 to put together a manuscript and get it published. So, you know, you can imagine a 5-year timeline being shortened to maybe 3 years. That's pretty fast. You've got to have all of your things lined up and the funding in place, of course, to do this, but this is certainly achievable. Yes. Yeah, so the question is, how does the tamoxifen drug induce the mutation in these mice? So this is a common genetic approach that people, experimental biologists, use in the mouse genetics field. And it's a genetic trick where the important element here is actually a bacterial recombinase that does homologous recombination in bacteria, but it also does it in mammalian cells when you express it in these cells. This recombinase is expressed in these mice, but its ability to function is controlled because the recombinase is fused to another protein, the estrogen receptor, and its ability to work is limited because the estrogen receptor pre-recombinase chimeric protein is held in the cytoplasm of the cells until you provide tamoxifen, and then it translocates into the nucleus, initiates recombination, and now you start expressing the gene of interest in this case. So there's nothing really untoward about the use of this particular estrogen antagonist in these mice to do these kinds of experiments. It's a common genetic approach that's used in the field. All right, thank you.
So for our next presentation, we have Dr. Jun Xie and Dr. Guangping Gao talking about the gene therapy project that we mentioned earlier and its current status, and we have a video for that. So we are going to try to pull that up. No, okay, one sec. We're going to work on getting a password and then trying to pull it up. And then Mac and Steph, we're going to get the agenda back on track. So you guys will be next following this video.
All right.
Good morning, everyone. I'm Jun Xie from Human 10 Medical School. I would like to thank the Yaya Foundation to give me the opportunity to present our going gene therapy project. Working with Dr. Gao, Dr. Bennett, we're developing a gene therapy product to treat post-related leukodystrophy using adeno-associated virus. Your gene-based therapy can be divided into two types based on how the medicine are given to the patient. One can give a patient directly like a shot. We call it in vivo. The medicine can be nucleic acid or viral vector carrying nucleic acid. Once the medicine was given to the human body, the virus or the nucleic acid will go to the cells with defects and fix them. Right now, the three viral vectors are used to treat human diseases. All of them are AAV vectors. The first one was developed and approved first in Europe as used to treat lipid metabolism defects. The second was developed to treat blindness. The third one is to treat muscle atrophy. Nucleic acid is also widely and rapidly developed to treat variable diseases. The second approach called ex vivo therapy. The cells was isolated from human patients, genetic modified in the lab, expanded, and put back into human patients. This approach is used to treat blood cancers with very high efficacy. Today, I will focus on AAV-based in vivo gene therapy. To develop a gene therapy product, 4 key components need to be considered. The first one is therapeutic gene. We need to know what gene we want in our cells, and we design therapeutic gene to fix them. The second one is vector, which is a vehicle to deliver the therapeutic gene in the disease-associated cell. The third one is that how we give the gene therapy product to human patients. we inject them into our blood vessel directly or into the disease organ. This all need to evaluate in animal models. The efficacy and safety need to be tested in animal models or large animals that can mimic the human disease. After all this process, the gene therapy product can be tested in humans, as we said, clinical trials. Vectors is the key for gene therapy. The 3 viral vectors. One is adenovirus, one is the lentivirus. The third one, AAV, we're going to use to treat the POL3 related leukodystrophy. The ideal feature for viral vectors is following. First, it needs to be very efficient. The second, we want one shot last forever. The third, we don't want the virus very high immunogenicity or it's toxic. Otherwise, our body will kick the virus out of our body. Then the therapeutic gene will be lost. The therapy outcome will be gone. And also the last one, we want the vector to be safe. We don't want the vector genome integrated into our genome, which will be unsafe. Adenovirus and lentivirus, they have their strengths, but with some drawbacks. Fortunately, AAV vectors have all these strengths, but no obviously drawbacks. It's very efficient. It's very safe. There's no known human disease related AAV virus. It can last forever, at least in mice. It lasts for its whole lifespan. And also AAV don't integrate in our genome, which render it very good safety profile. To engineer the wild type AAV genome into therapeutic region, 2 protein encoding gene wrap and cap were removed. Instead, the therapeutic set was incorporated into the virus genome. Only the packaging signal was still remaining in the therapeutic vector. They are serving as a packaging signal produced the AAV virus. AAV gene therapy is a teamwork. AAV capsid determine what cells they will get in. Right now, there are 9 major AAV capsids. They can transduce multiple tissues like muscle, liver, brain, heart, eyes, pancreas. Right now, still scientists are developing more AAV capsids for better efficacy and specificity into different cells. After AAV capsid gets in cells, it will release its genome as therapeutic product. Now let's assume the cells or the disease-related cells received the viral vector. If the disease is caused by a mutation that the gene are not functional, then the AAV vector can provide a correct and functional copy into the cell to supplement the protein the cells are missing. If the cells have the mutation which is toxic to the cell, then the AAV virus can deliver an inhibitor to silence the mutated gene expression. Then the toxicity to the gene, to the cell will be gone. By doing this, the cell will survive. And also, you may have heard gene editing. This is a very promising approach for future gene therapy. They can convert our mutated gene into correct version. Once we overcome the shortcomings of the low gene editing efficacy and over-targeting effects, we are very optimistic that the gene editing approach in gene therapy will be more promising. Except these 3 AAV vectors I mentioned at the very beginning. There are about, I think there are more than 100 AAV vectors are on the clinical trial. Those diseases are neurological disease, metabolic disorder, bleeding disorder, ocular gene defects, and muscular problem. Among all those diseases, CNS gene therapy draws the most attention because only AAV vector can transduce brain cells efficiently, not other vehicles or chemical. So the AAV vector make the gene therapy, make the CNS disease, which cannot be treatable, now become feasible. As you may know that the fourth AAV medicine that could treat AADC deficiency, which is also a brain disease, will be approved very soon in Europe. Then how we treat the POL3 related leukodystrophy. POL3 gene is deficient in our cells. That's we know. And also, it's reasonable if we can supplement a correct version of POL3 gene in the disease-related cells, we may be able to rescue the disease. Right now, we have construct the AAV genome that can express POL3 gene efficiently. This has been validated in the cell culture. Now we need to know what AAV capsid we can use to deliver this therapeutic gene. Right now, Dr. Bennett's lab is testing multiple AAV capsids in the mouse brain to figure out which capsid can transduce the disease-related cells the best. Once we know the cell type information, we'll produce the AAV vectors expressing this functional POL3 gene and test in the disease mouse model, which is available right now. I hope our team effort could bring a promise to the patient with POL3 deficiency. Thank you very much for your attention. And next, I want to thank the foundation and Dr. Guang Pinggao for his support.
So I want to thank Dr. Gao and Dr. Xie for their work and for their presentation. They're of course not here to take questions, but Valerie Greger, our science director, is. So maybe I'll pause and ask if anyone has questions regarding the gene therapy presentation. Aaron.
So, is this on? Yeah. So you said which method was most promising? So that depends on the disorder, and I think for our disorder, for POLR3 related disorder, it's replacement, and this is really Dr. Gao and Dr. Xi, their expertise, or one of their many ones, is really to construct the envelopes for the genes to be delivered to the cells that they should go to. Which doesn't actually quite answer your question, but and one of the take-home messages I wanted you to really have from his presentation, if you remember the picture with the pie charts,
of the tissues that can be targeted. This is a wide variety, and they're working on that you can, bring genes into the tissues where they are needed. I think this is one of them. And as I said, the question of whether you use gene replacement, gene editing, or other methods really depends on your mutations and your genes. And currently, for us, AAV replacement is most promising. It was a long answer. Yes. The question was which mouse model are used for the studies. And I think that Mark will speak more to that later, but it's Polar3B. It's the B mouse rather than the A mouse.
Yeah, that's actually good.
Yeah.
That's actually a great segue to our next presentation. So thank you for that question. And thank you, Valerie. So yeah, it's a great privilege now to introduce and welcome Dr. Mac Michelle Robinson and Dr. Stephanie Perrier, who's joining us virtually. So thank you, Mac. And are your slides here? Or could you give me a hand, Shannon? I'm not seeing them. Thank you.
Okay. Can you guys hear me with the mic? Great. Yeah. Okay, thank you. So to recap what Valerie said, I think with regards to the gene replacement question, one of the reasons, I just wanted to add one thing, one of the reasons that we're interested in that for therapeutic purposes is because, as you know, there are many mutations in the patient population, and addressing those through a gene editing approach requires us to address each of the individual mutations one at a time, whereas a gene replacement approach might be something that we could get to an endpoint faster. And of course, I think that's everybody's concern in the room is how do we get there as quickly as possible? So that's one of the reasons. And with respect to the model, like Dr. Willis, we also are working on animal models for the disease. And we're interested in Polar3B models because, as you know, that is a large fraction of the mutations in the patient population as well. So. That's there, great.
Can you hear me?
Yeah. Can you hear me?
Great. Yes.
Okay,
Thanks.
good stuff. So I'm going to jump in here. Some questions that we're all interested in. Why are we here, right? How long is it going to take to develop a therapy for 4-H leukodystrophy? How does drug development work? How realistic is a therapy? What kinds of research are we trying to accelerate? And what kinds of drugs are we working on? So we're all here today, including me, with questions like these. And so my name is Michael Robinson, and I'm joined today by Steph Perrier. And we work for Dr. Genevieve Bernard in the Myelin Gene Laboratory in Montreal, Quebec. And we study Polar3-related hypomyelinating leukodystrophy, or 4-H. And we wanted to discuss some of our thinking around the therapeutic approach to the disease. So we're aiming today to give you an overview of the drug development process and some of what I think at this point should be called major ongoing discussions in the field. And we want to talk about the kinds of research that we're working on and some of the treatments that are being considered for 4-H by ourselves and others, including both gene and cell-based therapies, which are areas of interest for our lab. And I think one of the themes of the talks over this last couple of days have been that we don't necessarily have concrete answers to all of these questions yet.
So we're just trying to open up a discussion about where things are going. And so feel free to chime in if you have questions. We'd love to answer them. So let's talk about the path to therapy development. What does drug development look like? And what kinds of questions are we trying to answer at each of the individual stages of drug development? So there are 4 stages to drug development. We have discovery and development, preclinical studies, clinical development, and post-marketing studies. Discovery and development is the earliest phase of research, and here we're concerned with really asking fundamental questions about how a disease works so that we can better understand how we could try and treat it. We might ask questions about mechanisms of disease, drug targets, or pathology, and in order to answer those questions, we develop models that allow us to study them in detail. And once we have models, we can also propose and test candidate drugs, which is something that we're trying to do. So the models we use come in 2 flavors. We have in vivo models where we study disease biology in a live animal. Sometimes we use a mouse, most commonly, but there are other animals as well. And in vitro, and that's where we use cells in a dish to try to study disease models as well. The next stage of research is called preclinical studies. And you may hear the term preclinical used loosely to describe kind of any research that comes before the clinical trial phase. But it actually refers to a set of experiments that we use to develop a clinical trials application. And in that particular instance, we have to usually have to follow some regulatory standards to carry out those studies. So in that case, what we're really doing is we're testing a candidate drug in our most useful disease models, and we're trying to figure out if we should proceed to clinical trials. So here, we're collecting data on a number of different outcomes, but 2 of the most important ones that factor into the decision about whether or not to proceed to trials are dosing and toxicity data. And as you can imagine, if you're in the shoes of a clinician, when you're starting a clinical trial, you need to know how to dose a drug and you need to know how safe it looks, right? Those are 2 very important factors. Clinical development is how we refer to the collection of clinical trials that are used to get a candidate approved, and really approved for use outside of the trial environment. So getting to this stage of research is a huge milestone, and it's arguably the most challenging of them all to complete. So here we want to determine if a drug is both safe and effective. And traditionally, clinical trials are split into 3 phases. And the first phases, sometimes phase 1 and 2 are condensed together. And we're asking questions about both of those phases at the same time. And the other important thing to realize is that oftentimes extensions can be added on to these different phases that prolong studies for additional years if there's reason to do that. So clinical trials are designed to ask progressively harder questions about how useful a drug is in order to know if it should be approved. 6 to 10% of candidate drugs are successful through all phases of clinical trials. And these numbers are sort of a rough guideline because it does vary a little bit from disease to disease. And the process is very highly regulated. So, I mean, you know about the FDA, we also have Health Canada, there are regulatory bodies in Europe and Japan. And, you know, these trials need to be regulated very heavily in order to ensure that they're conducted safely and that the data that we have is high quality. Once a drug is approved, we continue to do post-marketing studies. And there's a number of really good reasons to do this. The biggest one is probably that we want to continue to monitor adverse events and safety after a drug's been approved. But we may also be interested in trying to learn more about how a drug works, how effective it really is, and whether we can improve it or design a better one. So in order to describe kind of how our questions evolve throughout this drug development process, I wanted to pick on a science project that a lot of you would be more familiar with. So I picked the space race. Okay, so the space race took place over about a decade, starting in the 50s and throughout the 60s. And the big question was, can we land on the moon, right? It was a big question at the time. And you'll notice that when you start something like this, that's not the first question that you ask. So the first question that NASA asked was, can we put a person in space? And that was called Project Mercury. And that's NASA's Discovery and Development Project. So they sent 20 unmanned vehicles into space during that project. And they even sent 2 chimps named Ham and Enos into orbit before they sent 6 human pilots. And then they moved to Project Gemini. And they're stepping up the question a little bit. So they're trying to prove that they have the techniques to land on the moon. It's NASA's preclinical study, right? And they're asking, would the techniques that we have work for a moon landing? So in total, Gemini launches 2 unmanned rockets and 10 crewed flights. You notice that they're stepping up the question, but they're also committing more heavily to the technique. Most of the rockets are crewed at this point. The rockets are bigger, and they're testing out these techniques to try to work on a moon landing. So they prove that important things like spacewalks, rendezvous, docking, these are all possible in space. And at that point, things are picking up speed. But the one that everybody knows is Apollo. And you know the answer, or you know the rest of the story. There's footprints on the moon. So despite the odds and the incredible resources that this takes, it is possible to do incredible things. You may know less about NASA's post-marketing studies that's called Skylab. Skylab was instrumental in understanding long-term spaceflight. And ultimately, the research that was done set the stage for the International Space Station, the ISS that we have today. So I hope I'm painting like a more visual picture here of how our questions evolve throughout this process, right? And how the complexity and the resources required to answer these questions get stepped up at every phase as we move through the process. In the spirit of that analogy, I'm asking the question, are new drugs a moonshot? How realistic is it that we're able to produce a therapy for 4H? So firstly, we need to know that, like developing drugs and sending rockets to the moon, these types of projects take a long time. Half of all drugs approved between 2010 and 2020 spent longer in clinical development than the Apollo program took from the very beginning to when they landed on the moon. What's even more important here to remember is that the preceding programs that NASA pursued took a total of 10 years to complete. What that means for our understanding of drug development and the analogy that I'm trying to pursue here is that getting to clinical trials can take even longer than the trials themselves. The process of discovery is not trivial. On the other hand, it's also important to keep in mind in the world of science, that this is something that's done regularly. It's something that will continue to be done in the future, and we might even get better at it. We just can't predict how long it's going to take for a new drug to enter trials, and it's especially difficult to make an educated guess because the research that occurs leading up to trials isn't necessarily tracked the same way that trials research is. Optimistically, I think, though, rare disease research is growing. So orphan drugs are drugs that show promise for diseases that affect less than 200,000 people. And the data shows that since the 1980s, the number of orphan drugs being developed has doubled every decade. And with the help of groups like Yaya, we hope that to be the case for 4H as well. So more people working on rare diseases is important and is optimistic for all of us. We hope that it's going to mean an increased chance of a successful discovery and development effort. And we also hope that it's going to enhance our ability to conduct trials in a way that's more in line with the specific challenges inherent to developing drugs for rare disease. So in summary, there are 4 stages of drug development. We can't predict how long it's going to take to bring a new drug into clinical trials, and success in trials is never guaranteed. But there is more activity in the space than ever, and we believe it's possible, and we're working on it. So I'm going to pass it to Steph.
Great, thanks. So next slide. So now that we know that we've reviewed the process of drug development, we can discuss therapy approaches in the context of 4H leukodystrophy. Next slide. From studies of large cohorts of patients, as you know, we know that the 4 common cardinal clinical features associated with 4H leukodystrophy are hypomyelination of the brain, which leads to neurologic dysfunction, dental problems are involved, and endocrinology and growth abnormalities. And when we're looking at a therapy, we're looking at improving the neurodegeneration of the brain, so focusing in on the hypomyelination feature. Next slide. So before we can proceed with developing therapies, we have to gather more information about these features and how they progress over time in order to form a more full picture of the disease and how it evolves so that we can know how to treat it. And as Dr. Wolf and Dr. Bernard both talked about, this can be accomplished using natural history studies of the disease, similar to also how the Yaya Foundation spoke about it. So natural history studies are epidemiological studies that aim to collect information about a disease from a large group of patients so that we can identify the most common symptoms and see how the disease evolves, and also how often patients have mutations in specific genes. So the POL3 genes, POLR3A, 3B, 3K, and 1C. So this helps us form that full picture of the disease from the first diagnosis to the later stages of the disease. It helps us to identify biomarkers as well, which doctors can use to measure the stage of the disease and whether or not a therapy is working or not. So we can study genetic diseases in the lab using different kinds of representative models, like mouse models or cell models, like Dr. Willis spoke about before, mouse models. Next slide. So we can use mice to study human diseases because they can be genetically manipulated to mimic a disease or condition. So for example, in 4-H leukodystrophy, researchers like Dr. Ian Willis's group have created a mouse model to explore how mutations in POLR3A can impact brain development, while we're working on a model for POLR3B. So these models help us test therapies. They help us understand the biology of the disease and study myelination in a biologically relevant context. So we can also use cell models to study rare diseases, and this can be accomplished by, for example, collecting blood or skin cells from patients, growing them in a dish in the lab under different conditions, and using a specific method, we can use different growth conditions to change those patient cells into stem cells. And stem cells are just a type of cell that have the ability to turn into different cells. So you can see on the diagram here, once we revert them back to stem cells, they can change into neural cells or blood cells or pancreas cells. In this case, we're interested in studying the neural cells. And it's especially important to use stem cells, induced pluripotent stem cells is what they're called, to study brain diseases, because we normally can't study human brain cells without collecting them directly, which isn't possible to do. We can't perform a biopsy to collect patient cells because it's very invasive. So we can use these patient brain cells that have been derived from stem cells in order to test also whether certain drugs will work in correcting their dysfunction. So this is called drug screening, and it's very efficient using these cells in the lab because we can look at many different types of drugs, including some that have already been FDA approved, and test them at the same time. So it makes the workflow very efficient once we know what we're looking for and whether it can be improved or not. So I'm gonna, next slide, sorry, there's one more. So now we're gonna move on to discuss the different therapeutic approaches for 4-H, given what we've studied and also what has been trialed in other leukodystrophies and other rare genetic diseases. So Mac, we'll take over now.
Thanks, Steph. So now that you understand a little bit more about the research approaches that we're taking to answer questions about 4-H, I'm going to talk a little bit about the therapies that might be promising. But first, we need to review a little bit about the basics of genetic disease. So, as you know, genes are DNA instructions for cells. And your genes are kept as part of chromosomes inside the nucleus of every cell in your body, with the exception of red blood cells and platelets. So proteins carry out low-level functions in cells, and your cells put together the functions of these proteins in order to accomplish sort of higher-level things. And so they're like the building blocks of cell function. And your genome carries instructions for how to make protein. So in order to do this, the cell copies its DNA as an RNA method, and it sends that to the protein synthesis machinery to make a protein. That machinery is called the ribosome, and there are a lot of other associated factors there, but the message is brought to the ribosome, and the ribosome is able to, with the help of tRNA, interpret the code to make a protein. Because the correct synthesis of the protein depends on the DNA sequence, when you have DNA mutations, it can cause dysfunctional proteins by changing the way that those proteins are made. And so ultimately, that can sort of filter up and cause defects in cell functions, and ultimately it can lead to disease. Now, the process I described of copying DNA into RNA and using those instructions to make proteins is called the central dogma of biology. And luckily, we have therapeutic tools that we can use to try to correct cell functions at different levels of this process. And we're hoping that that's going to allow us to improve or correct cell function in the context of 4-H. So some of the ideas, and really this is sort of the landscape that we're working in right now. But again, these are ideas. This is the earliest phase possible of research. These are discussions at this point, and some things we've made some progress on and other things have yet to be tried. So this is really sort of my brainstorm slide, okay? So there is gene therapy where we would add healthy copies of POL3 genes to cells. There is cell therapy where you might replace defective cells in the brain and try to promote myelination with new cells that have been replaced. You could use small molecules to maximize the function of the existing POL3. You might try using RNA therapy to help produce healthy POL3 in the brain. Or you could use antisense oligonucleotides to help avoid splicing errors in the case of specific known mutations. So, as I say, ongoing discussion, but still very early days. So let's talk a little bit about gene-based therapies and what that means and what they are. In order to understand gene therapies, we need to understand how they're designed. So the first distinction when we administer a therapy is whether or not we're going to administer the therapy to the patient or to the patient's cells. So in vivo gene therapy is where we administer the therapy to the patient in the body. And ex vivo gene therapy is where we extract some cells from the body, we treat those cells in a dish, and then we re-administer them back to the patient, corrected. So in the case of in vivo gene therapy, that's done when the affected tissues in the disease are hard to access, and that includes the CNS or the brain, whereas ex vivo gene therapy is done in similar situations to where you could use a bone marrow transplant. And that includes blood and metabolic disorders as well as a handful of leukodystrophies. But in the case of 4-H, it's probably going to require an in vivo approach because we need to directly target cells that are in the brain and can't be removed. The next thing that differentiates therapies from one another is the method we use to get the gene therapy inside the cell. So you've heard from Guangping Gao and Dr. Junxi, and there are different viral tools that we can use to do this, and they're matched to the use case that we're developing for. So in vivo gene therapy has specific viruses that work well for that. Ex vivo has specific viruses that work well for that. We use viruses because they're especially good at getting inside cells. They're evolved to do that. But we engineer them, as has been alluded to, in order to remove viral genes so that the viruses can't replicate and spread inside the body. So what they are is little delivery vehicles. We're delivering a therapeutic gene to the cell, and we're taking advantage of the virus for that, for its ability to take the therapeutic gene and get it into the cell and get it into the nucleus. And as I said before, you really have to match the virus that you're using to the situation that you're trying to develop a drug for. So again, it has to be matched to in vivo or ex vivo use cases. But because these viruses have strengths and limitations like any other drug, it also has to be well matched to disease. The whole design holistically has to fit what you're trying to do. So it's very important that we test these viruses in our models, not just for their ability, not just for the proof of concept for the therapy, but also for the proof of concept that the virus is well matched for the purpose that we're trying to use it for. The gene therapy mechanism of action and the way that it works inside cells is another fundamental part of designing a therapy. So two common mechanisms are gene addition and gene editing, and you've heard of a couple other ones. In this case, I'm using gene addition to mean the same thing that Junxi meant by gene replacement. So what we're doing is we're adding a copy of a therapeutic gene, a normal, healthy, functioning gene, to cells that have the mutated copies in them. We're not replacing the copies that exist within the cells. In gene editing, what you're doing is you're adding genetic material as well as oftentimes an enzyme. So you may have heard of CRISPR-Cas9. And what that's accomplishing is it's allowing CRISPR-Cas9 to try to make repairs at the specific site where the mutation exists. So what would a 4-H gene therapy look like? So again, this is an idea that's garnered interest. It's something that we're currently interested in working on. Oh, no problem. Nope, no problem.
Thanks.
Okay. So we left off at what would a 4-H gene therapy look like? And the short answer to that is that it's a little bit too early to tell all of the details. It's most likely that if a gene therapy idea is feasible, that it's going to be an in vivo approach, as I alluded to before, and it's likely to require the use of a viral vector, so probably adeno-associated virus because of its specific strengths in the context of in vivo gene therapy. And it's also, I think, a strength that's been studied in a handful of other leukodystrophies, and it's being used now in a number of different trials for different diseases as well. So we know a lot about how AAV works inside the body in a clinical context. We're still answering fundamental questions about the feasibility of this approach for 4-H. So I think Jinshi and Guangping Gao alluded to the fact that we need to know, for instance, about the ability of the AAV capsid that we choose to target the cells of interest in the brain, in this case, cells in the oligodendrocyte lineage, the cells that make myelin. And so to this end, we've developed a mouse model of 4-H, the Polar3B model that we have now, and we intend to use this to test the gene therapy approach and potentially other approaches for treating 4-H leukodystrophy as well. And that work is currently underway. So I'm going to transfer over to Steph for her slides about cell therapy. Go ahead, Steph.
So we'll now discuss another type of therapy called cell-based therapies. So in the brain, we know that the white matter is a complex tissue. It's composed of many different cell types, as illustrated in this image. Next slide. So in the white matter, myelin is formed around neurons by cells called oligodendrocytes. In 4-H leukodystrophy, we know that myelination doesn't happen properly during development. And studies of brain tissue have shown that it's a complex disease, but we know that there's a loss of these oligodendrocytes that are available to make the myelin. So because the oligodendrocytes don't work properly to provide support to neurons, later on in the disease, parts of the neurons can also start to break down, leading to neurodegeneration. So if the oligodendrocytes can't myelinate properly, it's been explored in other cell types, whether they can be directly replaced by healthy cells from other sources. Because the oligodendrocytes are at a final stage and don't replicate, researchers have to explore whether precursor cells or cells that can turn into oligodendrocytes with time can be delivered to form the myelin properly. So in many studies of mice, researchers have shown for other diseases that delivering these pre-oligodendrocyte cells to the brain is successful in forming myelin. So this can be done by injecting these cells directly into the brain of the mouse and seeing if the myelin can form properly after a period of time. So this has been shown in other studies of leukodystrophies like Pelizaeus-Merzbacher disease, which is another hypomyelinating leukodystrophy. Next slide. So along these lines, a phase 1 clinical trial had been completed in patients with Pelizaeus-Merzbacher disease, again, another type of hypomyelinating leukodystrophy. And in this case, brain cells were injected into patient brains through 4 small holes in the skull, and patients were monitored over the course of 5 years. So the primary goal of this clinical trial was to figure out whether it was safe and whether it caused any unwanted effects. So after 5 years, the procedure was found to be well tolerated and no patients had formation of tumors, but 2 had an immune response. And in terms of looking at remyelination, and whether the therapy was working to improve the patient condition, there was less evidence that this was the case because they only saw very small changes in MRI at the injection sites, and there was only very little clinical improvement in the patient's conditions. So because this clinical trial was only done to assess the safety profile, the primary goal was met, but it still needs to be improved before it can be tried again as a therapy to see if it can work to improve clinical features. So even though some therapies can be successful in animal models like mouse models, it's important to remember that there are a lot of steps before they can be trialed in humans. And it's often difficult to translate results to humans when studying neurological disease therapies because mouse brains are a lot different than human brains in different ways. So first, they're a lot smaller than human brains. As you can see in the figure there, they're very tiny compared to our brain, as you know. And they also have a lot less white matter. So you can see why it would be less effective to try to transplant cells into the human brain compared to the mouse brain, because it's so much larger with more white matter that has to be repaired and myelinated. And also, mouse brains develop at a much faster rate than human brains. And while this is helpful when we study mouse brains in the lab, because we can see whether therapies can work on a much faster timeline, it takes a lot longer to translate that to humans to see if it works over a period of time. So that means that it takes a lot longer to develop these therapies to the point where they could be used to effectively in human diseases. So in summary, we talked today about both cell therapies and gene therapies in the context of 4-H leukodystrophies and which would be the most effective to consider studying in the lab based on what has been shown in other types of leukodystrophies and rare diseases. And if you're interested in reading more information in scientific detail about these therapies, we recently published a review paper with a reference at the bottom of the slide where we discuss different studies that showed results. And I'll let Mac finish with this slide here.
Okay, so takeaways from today's talk. The therapy development occurs in phases, and we're still in the very earliest phase of research for 4-H in terms of therapeutics development. I think it's optimistic that there's a lot of interest in this space, and we have partners like Yaya to help with this. And as Dr. Willis alluded to before, we're also leveraging decades of research on POL3, which is helpful to understand what it is that we're dealing with here. Another takeaway is that not all types of treatments are useful for all diseases, and that is doubly true for leukodystrophies. These are different diseases, and so we need to consider that success on one front does not mean success on all fronts all the time. As I said, we're still in an early phase, and I would like to take this opportunity to bring up that we've developed a brochure, and that brochure, the purpose of it is to try to help with conveying some information about these novel therapies. So we have information about in vivo and ex vivo gene therapy. We have information about ASOs. We also have information for other leukodystrophies. So we have information about enzyme replacement therapy as well. This is very new, and I'm still in the phase of collecting feedback, and we're trying to finalize things like illustrations and get this translated into multiple languages and things like that. So if there's anybody in the room or online that is interested in helping us with this, wants to take a look, provide feedback, I would really appreciate that. It would be super helpful to us so that we can get this done. And we'll try to provide that to the community through various channels like the ULF and Yaya in the near future. So thank you for your attention. And thank you to Ron and Valerie and Shannon and Christina for all their support and having us here to speak with you today. Okay, so the question is, has myelin been reproduced in the lab? And my question back to you would be, in what sense? Artificial myelin, or because my mice grow myelin every day? Okay, that's a fantastic question. So I think what you're getting at, and that really gets to the heart of what myelin is. So what myelin it's a component of cells. So cells are produced in the brain during early development, and they're produced sort of at the top end of the spinal cord, like in the brainstem, okay? And those cells populate the brain during early life, and they migrate to sites where they're needed to myelinate axons, and they extend processes, and those processes wrap around axons and ensheath them. So myelin is both a living structure and a highly organized structure. So what we can do in the lab is we can do things like we can extract myelin from a mouse or even from human tissue, and we can work with it to study things like immune responses to myelin, for instance, in the context of MS. But developing an artificial myelin substrate to put into a patient wouldn't work, because what we need the myelin to be alive. And that's what's difficult. And that's what Steph, I think, was alluding to about using precursors in a cell therapy approach, is that in that sense, what we would be doing is we would be giving the cells that would have myelinated the brain early in development. So we would try to recreate that developmental stage for those cells. We would implant those cells in the brain, and they would go and hopefully myelinate axons. And that's what they were trying to do in the phase 1 study with the PMD. But again, the interpretation of the effectiveness of that approach for the PMD study is complicated by the fact that they're really in phase 1, and that was they were looking at safety outcomes more than the effectiveness of the approach. Mm-hmm. So the microenvironment, sorry, yes. So the question was about the microenvironment, and I'm going to try to paraphrase the microenvironment of myelination. So the idea that in order to promote myelination in the context of, say, 4H or another leukodystrophy, we need cells to get to the correct location. They need to exist in a certain kind of microenvironment that facilitates myelination, and we need them to additionally myelinate axons. That is a very interesting concept and a difficult thing to address on a broad level. Different diseases promote different microenvironments within tissue, and some of those microenvironments are, in the context of disease, they're not permissive to myelination. So certainly that can be the case in MS lesions. That can be the case in other diseases as well. I think I'm trying to remember, but I think it was one of the EIF talks, and they had increases in precursors that they saw in tissue that was affected in the disease. So what that might reflect, for instance, is cells that are accumulating in a tissue that cannot go on to differentiate. And I think in Dr. Willis's mouse model, they've seen issues with differentiation, and in cell models as well, that's the case. So the microenvironment is certainly a factor and a challenge for promoting myelination in the context of hypomyelination or in remyelination studies. But again, the landscape is very complicated because each of these individual diseases has different considerations for what that microenvironment looks like. Does that kind of answer your question? Okay. Sure.
everything else can be done. But if you don't have the really understand and systems and the priority to do things that
Okay. So sure. Yeah. Okay. So to recap, the question is essentially, and again, I'm paraphrasing, but is it a fair statement to say that the natural history study is sort of the core factor that we need to address in the field? And can we move forward without it? I think that, I think no. I think the natural history is something that that needs to be done. It's something that I think a lot of people in this room and you know, key stakeholders and stuff are interested in and working on. Dr. Bernard may be able to speak to this more, but from my perspective, if I take a step back from my research niche and look at the whole field, I completely agree with you. Without knowing how patients evolve over time, it should be nearly impossible for us to design trials. We need to know how patients evolve over time in order to ask questions about how we would help them improve in a disease that occurs over many years. So you're absolutely right. And Dr. Bernard, I'm not sure if you want to add to that. She might have stepped out.
No, I'm here. So no, I don't have anything to add. You did an amazing job at answering the question, Mac.
Okay, great. What do we know so far is something that I think Dr. Bernard or Dr. Wolf would have better. I'm not a clinical expert, so I don't know. I'm not as familiar with that data as the research data. What I can say is that you're right. There's a substantial amount of variability within the patient population, and we do talk about, for instance, within our group, the idea that personalized therapies or more personalized therapies may become available. But I think that one of the things about that is that at the current time, that might not be the fastest way to get to a treatment for 4H. And so again, what we're doing is we're weighing what we think has the most potential in the space to move the fastest, that has a reasonable scientific basis, things like that. And that's what we're trying to focus on in the near term to try to make advances. And Dr. Bernard, do you want to talk about age or ethnicity in terms of how that might differ between patients?
I'm not sure I heard the question. Sorry, can you repeat it?
The question was about how factors like age or ethnicity might impact you know, the evolution of disease.
Yeah, so I think that if we classify the disorder by age of onset, the patients with a very early onset disease typically would, very onset, like 2 months, 3 months of age, which probably would be qualified as sort of like early infantile, if you want, or there's even some neonatal form. These patients will evolve much faster than the other patients. So the typical patients present as toddlers, and I guess the evolution varies according to many things, but the mutated genes play a role that we know. And the later onset patients that presents more with cognitive and behavioral manifestations often will remain stable for their motor development for several years before they start developing motor features. And then there are all of the other forms that Dr. Wolf talked about, the patients with no leukodystrophy, the patients that present with only with ataxia, only with spasticity. There are even patients that present only with bone disease or bone disease and ataxia, for example. So the variability is very wide, and that's why we absolutely need to study it. And regarding ethnicity, I don't think that we know whether or not it has an impact on disease progression. We know that some population have mutations in certain genes are more common in certain population. And we know, for example, in Quebec, we have a founder mutations for POA3A. So all French Canadian patients from Quebec at least carry one mutation, one copy of this specific mutation, but we don't know how it relates to disease progression. So I hope that answered the question.
Okay, so the follow-up question, Dr. Bernard, is, are we thinking that in terms of the therapies that we're developing, if we're going to try to make them available to all patients, or if they would be more appropriate for a subset like children under 5? Dr. Bernard, you're on mute, I believe.
Thank you. Sorry about that. So I think it's a very good question. We want to develop therapies for all patients. We don't know whether or not they will be benefiting all patients. There's a difference between when it's commercialized versus when it's tested. When it's tested with with the patient population, it's very important early on, if you want to assess efficacy, to include the right patients. So for example, if you include a patient that presents in adulthood and that has a very mild presentation and course of disease, if you give them therapy, you probably won't see an effect, unless, because you would have to do a trial for like 20 years or 15 years or something like that. So, and then no trials last for that long. You kind of have to do a trial for like 2 years, as usually the time that we have. So that's the first thing. And the other thing is, if you have a patient that is very severely affected at the time you try the therapy, and damage in the brain has already been done, the patient is less likely to benefit from therapy. So you really have to pick the right patient population, because one of the worst thing that could happen is that we would test a therapy that works, but would not be able to demonstrate it because we didn't pick the right therapy. But then once the therapy is available, there will probably be criteria put in place by different entities providing these therapies or paying for these therapies. But the patients that will be allowed to get that therapy will be it will be including more patients. But the first thing we need to do is prove that the therapy that we develop work. And then it's possible that it will be a combination of therapies, that it won't be only one therapy. It's the case for other rare diseases where there is, we know that, for example, one therapy works, but then another therapy works better, but perhaps one day both therapies will be combined and it will be even better. So it could be the same for 4-H, or maybe more than one therapy. And there may be therapies that are specific to a subtype of disease. For example, if we know that some patients have a mutation that is what we call a splice size variant. So it is close to the genetic material that codes into a protein, but it's not there. It's not in there yet. So it's possible that if we use ASOs, which are small pieces of RNA, to try to make this splice site work better to produce normal protein, we may improve the outcome of these patients. But of course, this type of very specific mutation therapy won't apply to anybody else but the patients that have this specific mutation. So the answer is complicated. I guess there's many different aspects to that answer, but I guess that these are some examples, I guess.
That's do you have a follow-up to that? Okay. Okay, I think that's it. So thank you very much, everybody.
Thanks, everyone.
Thank you.
Yeah, thank you. Okay, so we've blown way through our agenda. Clearly, there's been a ton to talk about this morning, and I want to thank everyone that presented this morning for all of your work and your partnership and your care for our community. So thank you all. Now we're still going to conclude at 12:30, but we have two presentations from the Yaya Foundation, and we are going to cover those. I think, Valerie, could you try to go sort of 10 minutes, and then Shannon, you take the rest, and then I'll do. Okay, perfect. And I'll do a rapid conclusion. So there was a question there about natural history and what's at the core. And what I'm really proud to be able to share is that we as a foundation have sort of formed a view on what we think is core. And we're not stopping there because so Valerie is going to be talking about our research priorities and our agenda. And Shannon is going to be talking about the work that she and Christina and Valerie are doing and we're doing to assess from the patient community, from all of us, what you all view as our needs, so that the work that Valerie has done is going to continually be informed by views from the patient community. So with that, Valerie, could I hand it off to you to talk about our research roadmap?
Does it work? So I will keep that short, and I will do this really for two slides that we have. And it has actually also been very well set up by Mac and Stephanie and Dr. Bernard and all of you, so we don't really need all this. What I want to tell you is, if I can get this to work. Which one? Okay. I want to tell you why we do what we do and how we are going to do that. So I hope that, I hope that when you come out here, you understand how we come to the things and how we prioritize what we do, which is what you asked. So what we want to do, of course, we want to understand what the disease is. We want to find a therapies and eventually a cure. And we want to do this and we want to do this accomplished by doing two things, which is bring everybody together in a room, which we did today. We have basic research, we have you, the patients, we have clinicians. So that's how we want to do it. So when I started out 2 years ago, and we were thinking about what we're going to do, we had all these things that we could do in order to develop therapies. So it's a lot. And so to take a step back, you've seen something similar from Mac's presentation. This is where we go when we develop a cure. We ask questions. We will ask basic questions in basic research. Do we have a pointer here? No, okay, never mind. We ask, thank you. We ask, what is the disease? How does it look like? How does it work? What does it do? Where are the patients? How can we identify people? These are the first questions that we need to ask. And then we can ask in the next stages, how can we tweak it? How can we change it? And then we come into what Mark had more described in preclinical. Is it,
Why is not in bed? Why is he got his earbuds in? Well, sometimes my fucking brain just goes.
Anyway,
I don't know what it is.
and
Brain
then we
keeps fucking
then we
going,
can
you know?
ask, is it safe to try it in humans? And then we go into clinical trials and post-trial monitoring. And what I also want to do, take you away from this slide, there is really three groups that need to be involved: the basic scientists, the clinicians, and you. And you have a function as patients at every single step of this way, and you have influence and you have power, and we need you. So this, when we sort of looked at that picture, where are we in 4-H? We are somewhere here. We have, as Ian told us, a good understanding of how polymerase III, what it does, but we don't quite know how to connect it to 4-H. Not yet. We have the gene, we have the mutations, we have some idea what the natural history is, but not really, as we know how we find. We have not found everybody yet. I don't think that we have found all the patients that are out there. So we are lacking there, but we have with gene therapy an approach that is at least promising to try. So I will show you with that thought, we did our first roadmap, and you know this, and I'm going to skip it, but this is when we wanted to do all. And we have in the meantime figured out that there's only 4 of us at the foundation. Somehow our clinicians don't seem to have more than 24 hours in the day, even though we try to do so, but so we need to focus. And these are really the 3 streams that we want to focus on. The first one, sort of the purple one, is really focused on patients understanding you. It's the natural history. We are trying very, very, very hard to get a natural history study underway, a more collective one. There are little ones out there, as you heard, or there are attempts, but we want to have something that is unified and that has that everybody can access, and that is really big, and where we collect everybody. So this is one of the things where you, as patients, you are the ones who are central to that. The other thing is, this is data collection and natural history. And the third one that we are just starting to think about is, how do we improve diagnosis? How do we make it easier for physicians to diagnose? How do we make sure that everybody knows about it? All these things. So that is sort of one block. The second one is the gene therapy program, and I won't go into that into detail. But that is another big thing that we're trying to fund. And as you have heard, we have some mice and some vector, and hopefully, if all goes well, then we can step through that. And the third thing is really that I realized we need to build our community. When I started, we have a good community of researchers, but we need more and more and more. And so this is the third thing that we're going to do, and hopefully that will spark work groups in all the other therapeutic approaches and the areas that we have not focused on yet. And that is really all I want to say for now. So if anybody has questions. Yes. Yeah. Mm-hmm. Yes. So the question was, yeah, I'm just gonna repeat, yeah, no, but it is, yes, I know where you're going. How about, if it's not that easy for you to go through this journey, if you don't have insurance, if you live somewhere where you don't have access to care, and things like this, and this is a huge thing, that there are resources out there. There are people who pay for genetic testing, and the question is how to gather these and we are trying to gather that information. And Ron, you wanted to, yeah, so there are, Rare Genomes is a project that can give access to sequencing, to whole genome sequencing. Invitae is trying to have studies where they test you for free, but you have to provide them your data. But there are ways to, well, we are starting to have ways to do something. Yes. Yeah. So, but I'm keenly aware of that. I just tried to. Any other questions? I know that, for instance, when you get to the point where you have drugs and where you have global companies coming in, Genzyme, or what is now Sanofi, had a big program providing access for patients to their medications, but we are not there yet. but yes. Of course, Genzyme is Genzyme, or was Genzyme. It was a company that I highly value. But anyway, so there are paths to do that, and people are thinking about it. So it is, but yeah. Well, with this, I think I'm going to turn it over to Shannon, who will tell you, now that I've told that if he had heard from us, yes, we're trying, but it's a long way out and you have to be patient. I'm sorry, I hate to say every time you have to be patient, but Shannon will tell you what we are doing, what can be done right now to get such a.
This work? Saving the non-science for last. Can everybody hear me? Okay. So I'm Shannon. I'm the advancement advisor at Yaya Foundation. I'm brand new. And basically what I'm trying to do is be part of this team and help us be sustainable, right? Because we want to be around to help you and new families that come around, either newly diagnosed or new faces that we find. You've seen this slide before. I think Ron has presented it before, so I'm not going to spend a lot of time on it, but obviously we're really, really committed in the next couple of years to help with research, right? To get to that next step. We want to provide support through these different methods. And that's my role at the end, right? Is we're gonna do a lot of different techniques to raise funds, right? And it doesn't happen in a vacuum. The thing about development and chasing money is I can't stress enough how important it is to have a really good understanding of the community that we serve. I've written hundreds of grants. I've done this a long time, and the more that we understand about the community, what you need, it just makes for a better story, and it makes it more compelling. And so we're going to work really hard on that. Okay. Great. So you guys know this. This is a really simplistic slide. But essentially, I just want to tell you what I'm doing, right? Like if you caught me on a Monday, what am I thinking about? These are the different things that I'm thinking about. So grants, we're going to submit grants, corporate sponsorships, like finding companies and different folks that are interested in what we're doing, that share our commitment to this community. Major gifts, so that could be individuals, that could be lots of different people, and then third-party events. So that's when a family or a community member that's interested in helping, maybe they hold an event in their community. So we've got Kurt in the back. This is something he would kind of fall into this category. We'll do a plug for 444H. He's doing a golf out. Stand up, my friend. Yes. If you don't know it, 7 years. Yeah. And Kurt is a really good friend to the Yaya Foundation and a co-founder. So it's just a nice way for him to get his community involved and to support him and his family, and also the greater good of this community. So I know that families get hit a lot for fill out this survey, do this registry. Here's another one. We'd love to hear more from you. And I get that that's exhausting. But I'm going to ask you if you might think about doing another one, because what we want to do at the Yaya Foundation is we don't want to create programs, initiatives, all of those words in a vacuum. I want to know from you what you need and what you're interested in. And that might be really different, if you have a teenager, if you're an adult, if you have a child, if you feel like you live somewhere that you don't have a lot of support, or maybe you live somewhere and you do have a ton of support and you want to help somebody else. So this is really important to kind of drive our next, we're almost at the end of 22, but thinking about 23 and what we can do to help you, because that's why we exist, right? Ron and Christina, since I've started, I'm so impressed with their commitment to the community and really listening to what you want. And so I'm going to ask Lindsay if you wouldn't mind dropping the link in the chat, and we'll email it out. It's a Google form. It isn't fancy. It's only 4 pages, 25 questions-ish. But some of the reasons, you'll see questions on there about the conference. What do you want to do for the conference? Do you want to have a conference? Do you want weekly calls? Do you want to have meetups? Do you want programs for kids? What can we do to help? And that will help us kind of target what we want to do, because right now it's like an ocean, right? We're all swimming in it. Like, we could do this, we could do that, we could do this, but I want to be really targeted in that, and then go after funding to help support that, right? So those are the things that we're thinking about. There are some questions about, like, where do you live? Like, do you live in a rural area? Do you live in a city? And the only reason that I'm asking that, to be transparent, is just to inform development work. Sometimes grants are available that might be like, oh, we only give in cities, or we only give on the western side of the country, or we only give in Argentina. So I want to know those things so I can go after them and be like, actually, yeah, we do have families there, or we do have need there. So that's why I'm asking it. It won't be shared with anyone. It's just existing for us to kind of drive what we're doing and find the right folks to help us do it. That's my presentation. Any questions? No? Oh, do you have a question? I'm sorry. Do you have a question? Yes. Items? Yeah. So the question was, there was a silent auction last night. Could we help donate to that next year? And I think the answer is yes. I think people will absolutely take that. And then to take it a step further, so yes, for ULF, that would be amazing. And then also at Yaya Foundation, do we want to do one, right? Like, should we be doing one within the 4-H community? So I'll be talking to you. And yeah, so I think that there's like both ways, like we're trying to help the greater leukodystrophy community, and then also think about the 4-H community as well. But absolutely, if you have ideas, I'm open, and I'd love to chat about it. Yes. Absolutely. So the question was, if you find something like a grant or an opportunity, can you send it to me? And please do, because I'm one person and I am like combing through these in the dark of the night. So please feel free to send anything that comes away, and maybe it won't work, but maybe it will. So it's worth it. Okay. Awesome. Thanks.
Okay, so I want to wrap us up. I'm going to do it quickly. Thank you everyone for sitting through a long morning of presentations, but I love the enthusiasm. And okay, first of all, I've never seen Valerie and Shannon present in person, right? And so I used to play basketball. Every once in a while, coach would do like a motivational speech and you'd feel ready to go. And when Valerie, you were presenting about from this to this to this, I was sort of like feeling really ready to go. And I hope you all were too. So I've never seen this slide before, but like Florian, we had breakfast here 3 years ago. If someone would have taken a brain scan of my brain during that breakfast, this is what it would have looked like. Just a jumble of stuff everywhere, not knowing where to focus, where to invest, where to give attention. Okay. And now fast forward 3 years and look at this. I'm like, Valerie, this is work that we should be really proud of. These are our focuses, and that is progress. That is progress. We are a small patient community. The idea 3 years ago was to prioritize and then invest where we in the few areas that were critically important and where we thought we could make a difference. And we're doing that now. And that's progress. And I see this and sort of think back to a few years ago. And I think forward to where we're going to be in 3 years. And it's very exciting. And so we can yes, Marcy was there, Claudette was there. So it's thank you. It is amazing. And I want us all to leave this room thinking about where we're going to be in 1 year, in 2 years, and 3 years. And I hope that what you all take away from this meeting is that we can only do it together. And by together, I mean us as patients and families, us as researchers and clinicians working together. We can only do it together. So when we ask you to fill out surveys, I've filled out a lot of surveys. They're super annoying, but they're critically important, right? Like our researchers don't know what we are experiencing unless we tell them. We don't know what you all find as the most important things unless you tell us. So please do take the time to fill out Shannon's survey, our survey. If you haven't yet enrolled in our data collection program, like, look, participating in research is a highly personal decision. And I don't want to tell you, I can't tell you what to do, but consider doing it or do it. It's really important. And when we tell industry or researchers, hey, we have 65 people in our data collection program, it's less than a year old, we see eyebrows raised, right? And if we can get to 100 and to 200, those are numbers that are commercially viable for industry. And at the same time, we're de-risking therapies, we hope, such that pretty soon we'll be at a point where we'll need industry investment. And if we can show that A, we've de-risked stuff, and B, we have a patient community that is willing to fill out surveys and participate in research, and we know who they are and we know where they are, that's how we're going to make progress in our disease area. So I hope we all leave this room excited about the distance that we've traveled, excited about how far we're going to travel together in the next few years, and really committed to doing this together. So thank you, everyone.